still his interest lies in basically the area of microRNA and also long coding RNA, and he is still actively working in this area. For his contribution to his research and also to the computational tools to analyze genomic data, he was conferred the CSIR Young Scientist Award for Biological Sciences by Prime Minister of India in September 2012. And I think is one of the most apt person to talk about this particular topic on precision medicine, and particularly the emerging technologies in the human healthcare. So now I call upon Dr. Vinod Skani to give his talk.
So good afternoon, everybody. So before I start my talk, let me first introduce what my lab works on. My lab works on this interesting area called genome informatics. That necessarily means that we don't do any experiments. We work on data and we analyze data to understand biological processes. And when we say genome informatics, it essentially means that we work on genome scale data. The many themes that we work on in the lab, most of my lab really works on the area of vertebrate genome organization and the regulation, including epigenetics. The other part of my lab largely works on the interface between clinical sciences, or clinical medicine, and genomics, where we try to develop techniques and tools to help clinicians to arrive at a diagnosis much earlier and in many times much more precise. Before I start my talk, let me first introduce how we landed up where we are. We started a lab way back in 2006, and as you see here, this is also a time when the cost of whole genome sequencing was dwindling so rapidly that it is almost a hypothesis that practically in 20 years from then, people would sequence their whole genomes. Right? They have not reached there yet, but yes, they have reached an affordable level where a lot of people can at least do part or whole of their genomes. But rather than the problem of doing a whole genome at that point of time, was largely addressing the gap in the knowledge about whole genomes. So in 2000, so this is a slide from 2008, this was the whole genomes that were sequenced at that point of time. This includes the reference genome, the London, the Watson genome, and the Chinese, Japanese, and the Korean genomes. Right? As you see here, these are the whole genomes that were available at that point of time, and this is where the whole genomes were derived from. So the top panel is nothing but an admixture panel of the entire world, and they're sort of sorted by the most recent to the oldest populations out there and how they are admixed with each other. And as you see here, actually all the genomes that were done at that point of time were from quite homogeneous populations or less admixed populations, as you see here. This includes the African genomes, the Chinese, Japanese, and the Korean genomes from East Asia, and the London Watson genome, which is from Asia. But as you see here, the largest admixed populations are in this part of the world, which is the Central and Middle East Asia, for which we do not have a representative genome, or we don't really understand what contributes to this admixture. And that's when we started our journey to actually start doing whole genomes in this country. This is way back in 2008. These sequences don't exist anymore. But in almost a year's time, we put together a draft whole genome from India. More than putting the draft whole genome, what is more important was how do we mine this data in terms of understanding very specific parameters, which includes variants which are of clinical importance, variants which are of research importance, and of course, be able to help clinicians to do analysis and interpretation. Well, that seems to be a very, very, what you call, large or crazy idea at that point of time. So we didn't really stop with doing the first Indian genome. We also did the first Malaysian, the first Sri Lankan, the first Philippines, and we also were one of the core members who formed the 2 Pan-Asia Population Genomics Initiative, which aims to sequence or understand the genetic diversity of entire Asia, or Southeast Asia in example. We also went ahead and forged collaborations with Middle East. Now we have collaborations spanning almost all these countries around this region, including looking at disease samples. We also host one of the largest variants in this particular area, databases in this particular area, which is called Almina, which encompasses almost close to 2,000 whole genomes and exomes. Now the real question is, of course, you can do genomics, you can analyze this data, put it in your databases. How can you really put this into clinical application? And that's where the question of an average patient comes. It's a sort of an oxymoron, because when we talk about personalized medicine, we talk about an individual who has a disease, his genome, which is very specific, and a targeted therapy which can actually ameliorate the phenotype or the disease in that particular patient. But in almost all practical senses, what in modern medicine we do is actually treat the patient as an average patient. And I say an average patient, which essentially means that almost all the clinical trials done in modern medicine are built on groups of people, right? Groups of cases and groups of control. And we tend to almost always consider what is the mean differences between these two groups. So in other words, we are almost always treating or developing therapeutics for an average patient. An average patient who is representative for that particular, right? Which is quite contrary to what the forefathers of modern medicine have originally stated. He originally stated in his book, and the statement in quote is, it's far more important to know what person the disease has than what disease the person has. Right? But what we really practice in modern medicine, and what I learned 4.5 years in modern medicine, is essentially to treat the disease and not really the patient. Now, the problem with treating such patients in an average phenomenon is that once in a while you get something like this. This is one of the most severe forms of adverse drug reaction. It is known as Stevens-Johnson syndrome. Of course, adverse drug reactions could be of multiple different severities, type A to type F, and of course could vary in the morbidity, in the mortality, and in the types of drugs that can cause these type of adverse drug reactions. In other words, what we need is to have a modern system which we call as genomic medicine. And genomic medicine is nothing but how can we improve the medical practice that we do today by integrating the genomic information, by trying to understand are there genomic markers which can make disease diagnosis and treatment much more precise, and how to integrate that into real medical practice. Now, when you talk about adverse drug reactions, you would almost always think that this is way off knowledge, which is not really. And probably one of the only studies, epidemiological studies from India, this is estimated to be approximately around 4% of people who are admitted to an emergency department. And 4% of people admitted to an emergency department, taking the whole population of India, is a very, very huge number, right? And on an average, each of this patient spends around 6,000 rupees just because of adverse drug reactions, extra to the cost of medication on. Now, to understand and prevent this adverse drug reactions, you need to understand how the system works. And to understand how the system works, it has to be in a simplified form. So, in many simplistic ways, you can describe the system as the gastrointestinal tract, for which you take the medicines. Of course, you can take the medicines through the system circulation on the parenteral route, and in some cases through inhalation, so it has a lungs here. And anything that gets into here is first metabolized by the liver, which is called the first pass metabolism. And whatever is metabolized, either from an active to an inactive form or an active to an active form, is pushed back into the systemic circulation. Now, what happens in systemic circulation? There are two organs. One is the lungs and the liver and the kidneys. The kidneys would excrete all the water-soluble molecules back into the urine, and which is the way of excreting this xenopathic molecules. And a set of molecules which can also be excreted through breath could be excreted back into the air. So in very simplistic ways, you can take the drugs through this route, you can take the drug directly through this route, you can take the drug directly through this route. In any of these ways, you take the drug, eventually it gets redistributed in this particular space, which is called the extracellular space or the system. Now, to make this even much more simpler in terms of genomics, you have the drug, you have the drug target. So the drug target is where the drug interacts and causes its effect. Before the drug can interact with this drug target, it has to cross multiple membranes. The multiple membranes could be crossed by either the active transport, using carriers or transporters, or inactively by just permeating through the membrane. And whatever permeates through this membrane could be either metabolized by the liver or the kidneys, or excreted largely by the kidneys or the lungs. And whatever is left after this is what is available for interaction, and which in interacts with the drug target and therefore causes the effect. So if you really go back and answer this question, is there enough evidence to put genomics in a clinical practice? It comes to a very vibrant field which is called pharmacogenomics, which today grows at the rate of around 1,200 publications every year, both in terms of the volume as well as in terms of the total contribution to the publication. This is approximately 0.09% of all the publications that are published around this world. But of course, putting this information back into a very systematic and organized form is going to take a lot of time. And that's where we formulated this sort of open source collaboration, which is called OpenPGX, which involved very large number of students and researchers and clinicians to be able to create an open access data resource, which will pull in all this information back into a very systematic format to build software which can interpret this information and put it back into the whole genome or the whole genome sequence. And of course, to be able to formulate evidence-based guidelines for testing, genetic testing, as well as for integration of the testing into clinical applications. Of course, this data goes through multiple different rounds of curation in terms of theories for relevance, in terms of applicability, and so on. I'll not delve deep into it. But in summary, what we could build is a very complex network, like what you see here. To make this very complex network simple, you have all the drug therapeutics here, you have all the drug targets in here, and before the drugs can reach the target, it has to cross the membranes and the transporters, or it has to be metabolized from an active to inactive or an active to inactive. So each of these molecules chart their own path. Many of them would not have a transporter or a metabolized enzyme. They could directly interact with the targets. So you could chart each of these molecules, around 6,000 odd molecules which are either in market or in investigation, to their drug targets by putting them across one of these layers. Now, of course, it's impossible for anybody to understand what these drugs are, what these targets are, and how they are going to interact. So you need to put this information into a very, what we call, simplistic form, which can be understood mostly by clinicians. Of course, even I cannot understand this thing. So that's what we try to do, is to actually put them into a software framework where you can actually put in a whole genome sequence or a part of whole genome sequence, which is called influence sequence. Put this information layer on top of the whole genome sequence in terms of the variations. Try to interpret it and provide an interpretation for a particular individual. And to make this visually more appealing, what you could do is you could create roadmaps like this. So this is very similar to the roadmaps like moving from Gurgaon to Noida. You have to go scroll 2 or 3 toll booths. So you have the drugs in here, or the drug categories in here. You have the transporters, the metabolizing enzymes, and the drug targets. So if you have a mutation which can prevent the transport or the metabolism, you would have a colored line on there. That means that there is a roadblock. But if you have a dark bar over here, that means that there is no roadblock, which means that the drug can cross this transporter, can be metabolized by this particular metabolizing enzyme and then reach the particular target. But of course, you will see subclasses of drugs which are either transported or metabolized by an inactive form of a particular gene, which is metabolized by an inactive form of another particular gene. So the effective level or concentration that will reach the particular target is either high or low, depending upon what the directionality of this transport and metabolism. So in other words, you can follow each of these drugs, which are lines, and try to see whether they follow through a black line or a go phenomenon, or go to a colored bar, which necessarily means that there is a roadblock. This is for one single individual. Now, of course, you could build this for hundreds of individuals, and that's what we did for the Malaysian genome, and not delve deep into it. But I'll delve deep into 2 such drugs which have very, very high genetic effects. One is warfarin and the other one is clopidogrel. Both of them have been extensively studied in literature, and practically this causes a very large problem to elderly people. As estimates suggest, around 33% of elderly people associated with hospitalizations, and 13% of elderly people who go to a hospital because of adverse reaction are because of these 2 drugs, warfarin and clopidogrel. The therapeutic range of these 2 drugs are extremely low. That means that you need to really titrate the doses. And the genetic variability also is extremely large, which is approximately around 16 to 20%. Now the idea of studying these 2 molecules is that since they are also widely used in the population, practically anybody who has a cardiac event or a cardiac stenting procedure takes either of these 2 drugs for their cardiacs. Right? So there's a very large number of people who already take these molecules. And in fact, the adverse reactions to these molecules are very well known. And that was also the time when we were looking at large-scale population genomes. And in this particular case, we put together one of the largest population-scale genome databases for Middle East and North Africa, which is called Almina, putting together around 2,000 whole genomes and proteins. So the idea is, can we apply this information onto a population-scale database and now try to understand what is going to happen for this population, or can you predict what is the population going to respond like? There is Carl here. There is Carl, yeah, it's there. So there is Carl's work, which is part of her end of thesis. So she took one of these populations from this database, which is the Qatar population, and tried to understand what are the variants which are associated with porphyrin or purple effect in the Qatar populations, and of course try to see within the populations and subpopulations are there differences. So what we found is that we have a high prevalence of high dose associated variant, right, and a low occurrence of purple resistance. That would mean that the dosage for these drugs need to be titrated very effectively in this population, because there is a very high prevalence of a high dose associated variant. Or in other words, most of the people who are taking this particular molecule, which is warfarin, would be probably getting in the standardized dose a lower than required dose for this particular. And what we interestingly found is that most of this variability were attributed to one of the subpopulations of Qatar, which is called the Bedouin populations, which accounts for around 70%. Of course, you could do that in India, and that's what we did as part of the Indigo consortium. So this is a cross-sectional consortium where the genome association studies were done for approximately around 2,000 individuals from North India, 5 states, which includes Delhi, Haryana, Punjab, UP, and Bihar. What I show here is the allele frequencies of these variants associated with warfarin and clopidogrel. In this particular case, this is clopidogrel. We have another one for warfarin. If you know clopidogrel is actually taken in as an inactive molecule. It's intestinal absorbed. It undergoes bioactivation through specific enzymes, and then causes inhibition of platelet aggregation by acting on the very specific gene, which is P2R1. Now, if you plot the allele frequencies of these genetic variants across the different populations, in the bottom panel you have the Indian population and the average of the North Indian population. This is the populations which are part of the 1,000 genome consortium, and this is the population which are part of the You don't really require to do p-value testing to suggest that the allele frequencies of North Indian population is quite distinct from the rest of the world. And what these allele frequencies would mean is that we have very good absorption, very good bioactivation, and very good inhibition of platelet aggregation, which would otherwise mean that the standard dosage prescribed for clopidogrel, which was based on the Caucasian population, is probably much higher than what is required for our population in North India. We have now tried to extend this in the southern part of India, and we have very exciting research for it. Probably next time we should share that. Now, while we were talking about genetic variants, there's a very important distinction that we need to make for genetic variants. So, genetic variants largely can be categorized into how frequent they are in the population and what are their effect sizes. Effect sizes would mean that what is the penetrance of that particular genetic variant. Or in other words, if I have a genetic variant, what is the chance that I will have that particular phenotype which is clinically relevant. So most of my talk till now focused on very frequent variants in terms that I put a cutoff frequency which is 1% of the population. Most of these variants are very low effect sizes, or in other words, if you have a particular variant, very unlikely or there is not 100% surety that we will have that particular disease. So these variants and these diseases include diseases that we commonly see, which includes diabetes, obesity, and so on. Right? But on the other hand, we have variants which are extremely rare in the population, probably 1 in 1,000 or 1 in 10,000, but have a very high effect size. Or in other words, if you have a particular variant, we are absolutely sure to have a particular disease, right? Or you have a very high penetrance. The ways and means to study these two diseases and methods are also quite distinct from each other. But all I talked about is variants which are moderately frequent in the population and very moderate effect sizes, which include pharmacodynamic proteins. What I have not talked to is common diseases and common variants, which include diabetes and cardiovascular diseases. What I am going to talk in the next set of slides are set of these variants, which are extremely rare genetic variants, rare in the population, which cause diseases with very high effect. So in other words, if you have this variants, you can probably predict that you are going to get this disease, or you already have it. The methods to understand these two kinds of disease variants are also quite distinct. For the common diseases, you use something called genome association study, and for the rare genetic variants, you use a term which is called whole exome or the whole genome sequencing. So, coming to the rare genetic variants have been known for many years now, probably centuries now. They were usually marked as monsters, right? And this is a pictorial description of the monster of the allele. And to put this into context, these are nothing but genetic disorders which are caused by mutations in the genome. And in many of these cases, they have a name associated with this, and this particular monster is otherwise in medical balance known as a Wolff syndrome. So, while we think that this was in olden times when people didn't know about genetics, of course, genetics was not known in terms of disease-causing genes, we might think that it's much better now, which is not really. In 2015, we are not really much different. You see paper clippings, this is from 2015, where for this particular example from very nearby Agra, the parent wants to euthanize all the 6 kids suffering from genetic disorder because they don't know what the genetic disorder is, and they are sort of an outcast in the community because it's something like a bad omen to happen to them in that. Now the real question is, how can we solve this? And to solve this, of course, since they are extremely rare, we need to have a very large network, and that's what we created as part of what we call as the Guardian Consortium. Today the Guardian Consortium has almost around 150 clinical collaborators from all across the country, from over 40 major centers, clinical research centers, and probably making it one of the largest consortiums for rare genetic diseases in not just in India but the whole of Asia. So what we do as part of this consortium is we have clinicians who see their occurrences of genetic diseases. We put them through a standard pipeline of whole exome or whole genome sequencing. We find the variant and report it back to the clinician who gives it as a report and advises the patient on what to do about it. And once in a while we get very new genetic variants and very new genes which cause genetic diseases, and that's what the research that we do. For more information, you can actually go back to the website. So why do we need to study rare genetic diseases? It's very important because India is a very large population. Of 300 million people who have a rare genetic disease in the entire world, India is home to 70 million. And by definition, anything which has an incidence of less than 1 in 2,500 is a rare genetic disease. So anything that you can think of, thalassemia, which is sickle cell anemia, they are rare genetic. The opportunity in India is that India is a very large country, so practically any genetic disease that you can ever think of, and that you can ever describe in literature, you have probably dozens, if not scores of them. The bad part of this is people do not know that they exist. The second good thing is that we have very large family structure and there is traceability. Practically everybody in this room knows who their grandfathers are, who their great aunts are, where did their family come from, etc. Which is a very good starting point to understand genetic diseases, because genetic diseases need to be known in terms of the pedigrees, and the family structure is very important to estimate what the genetic diseases are and where they come from. So the idea was, can we put a platform to be able to analyze genetic diseases in the country, because there is no statistics available for any of these diseases. Can we diagnose them appropriately using molecular tools? And of course, can we do research in terms of identifying the variants as well as the genes and what is the biology behind this entire program? There is a very robust database, because if you want to understand what is rare, you need to understand what is common. Right? So we put together from a large number of collaborators, a very formidable number of exomes and genomes, around 1,300 of them, into a database which encompasses practically the diversity of India. And this is the database onto which we query each of the genetic variants and see whether they are rare or whether they are not. To put a few examples, these are the kind of patients that come to us, very large families, very large number of people affected, and this particular case is a skin disorder where the skin erupts into burn-like appearances all over their body. It's not deadly, but it's disfiguring. This large family came from Gujarat. We did a whole exome sequencing of 2 individuals, found the variation, and of course go back and validate this entire. So this is a very fertile ground to understand the genetic variants which run in very large families, and to identify novel variants which causes genetic diseases. The good part of identifying such novel variants is that you can take this particular variant, go back to the community and ask this question: for any people who have this particular kind of appearance, you can test for this particular genetic variant which becomes extremely cheap, around 500 rupees, and now be able to provide a diagnosis at a community level and at a society. Of course, there are much severe genetic diseases. Of course, there is also the skin, where the skin erupts into such malformations all across the body. Same way we did exome sequencing, identified the variant, and the application of identifying such a variant is that now you will be able to prenatal testing and be able to prevent the disease from being occurring in the next generation. In other words, you can put a full stop to this genetic disease because you exactly know what is the genetic variation which causes the disease. And in this particular family, we have a perfectly normal thing was born out of the prenatal disease. Coming back to the original family from Nagra, well, this family had the oldest kid in this family is affected kid is approximately 12 years. In other words, this family has been suffering for 12 years without a molecular diagnosis. And without a molecular diagnosis, you do not have a perfect or a targeted therapy. And if you do not have targeted therapy, there's pretty much nothing that you can do with this family. Okay? So the first point is you need to have diagnosis, perfectly molecular diagnosis, and with the molecular diagnosis, you should be able to treat the kids appropriately. Now coming back to this original family, we did whole exome sequencing for this particular family. We identified a perfectly novel genetic mutation in the MLC1 gene. MLC1 gene was previously associated with megalencephalonic leukoencephalopathy. It's a cortical cyst type 1, extremely rare genetic disorder which occurs 1 in 100,000. Right? But you have a family of 6 individuals who are affected with this particular disorder. Now, what do you do with this? Of course, you have made a diagnosis, and with the diagnosis, what you can do is you can now go back to the community and screen for individuals in the community who are likely to get affected with this particular genetic disorder or are carriers for this particular genetic disorder. By understanding who is the carrier, you will be able to advise them that don't marry between the community. As you see here, is a very endogamous community. Practically every uncle marries within the family. And this is not very uncommon in this part of the world. What we found was very stark. With this particular variant, which we did a screening of approximately 84 individuals, around 28% of the individuals in that particular community, which is called the Nalban community, were carriers for diseases. That means that they do not have the disease, but they have one allele which is abnormal. They marry another person within the same community, they have 1 in 4 chance of getting the disease. By screening this entire community, we additionally identified another 3 more families with this genetic disease, which was previously not known. Right? Kids with this genetic disease are confined to their houses and nobody really knows, even within the community, nobody really knows that they have the disease. So what we have today is, from whole exome sequencing of a family, we have a genetic variant which we know that causes the disease. Of course, at a very high carrier frequency rate of around 28%, which would approximately translate in terms of statistics, 1 in every 100 kids who would be born in this community would have this particular genetic disorder. And that's how huge that impact is. And now we have a genetic test which we can roll out for approximately around 500 rupees or even less. In many cases, we can roll it for free to be able to do carrier detection, so that we can go to the community and tell couples that both of you are carriers, probably you should do a prenatal test, or we should tell them that both of you are not carriers, you can go ahead with the pregnancy. We can diagnose an additional number of people from this community, and of course, much more than that, as you can prevent this disease from occurring in the next generation. So, in other words, you can put a full stop to this particular disease from occurring in the next generation. So, to implement that, what we have created is a platform which we call as GOMED, which stands for Genomics and Other Omics Technologies for Enabling Medical Decisions. So we have 2 platforms. One is Guardian, where we discover genetic variants, and once you have discovered genetic variants, you need to convert this into a cheap assay which needs to be running in the community for thousands of individuals, and that's what GOMED does. And this is a very recent program which is almost a year old, less than a year old. We have catered to close to around 2,000 people, with around 80 to 100 high-end gene tests. We are expanding this program much larger to cover the entire part of the country. In other words, what we have estimated is that for every single family whom we have offered a genetic test and we can prevent the disease from occurring in the next generation, we have saved the society approximately 50 lakhs per year at the cost of a few hundred rupees. But that needs to have a lot of work to be done in terms of understanding the genetic makeup. Well, the ultimate aim or the ultimate goal is not really to just offer genetic diagnosis. Genetic diagnosis is an important component of it, but is to understand how did the disease occur, what is the biology of the disease, and of course, how can we treat the disease. Because in all the cases that I described, we can only offer a diagnosis, not really offer a treatment of the disease, because in many cases there is no treatment available. So the ultimate idea is, can we understand these genetic diseases in a model system, and the model system that we use is zebrafish. We can make the exact same genetic modification in zebrafish, either by putting in the human gene or mutating the native zebrafish gene, and now be able to screen for small molecules which can eliminate different types. In other words, what we think is a new paradigm for genetic diseases, where individualized fishes would be available for people who are suffering with genetic disease, and you will have molecular screening platforms where you can screen for the genetic diseases or genetic variants on a very, very large scale. We have some early successes for this particular program, and of course this largely relies on a very large clinical collaborative network as well as a student network, mostly students like you, who contribute to building computational models based on machine learning to be able to look at molecules which can perfectly bind to mutant proteins and can undo it. Before I stop, coming back to the original question, if this is all possible, then why is it not possible on a very, very large scale? And the real question, the real answer to that problem is that we do not have enough people to make sense of it. It's much easy today to make data available. It's much easy to sequence genomes. In a lab like IJB, you can sequence a few hundred genomes every week. Right? But there's not enough people or qualified mathematicians to be able to make sense of this data, because in many cases this data needs to be looked at much bigger depth. Of course, we try to automate a lot of these processes by building very specific clinically oriented databases and very specific software which can automate a lot of this analysis, but not of course all of it. One of the software which we have built for mitochondrial diseases is now commercially available, which has brought the cost of genetic diagnosis for mitochondrial disorders by almost 1/50. So that necessarily means that by automating this entire process and making it much more accurate, you can get rid of a lot of manpower, which is expensive, and make the diagnosis much affordable and of course in a very, very large scale. So this particular software can enable mitochondrial genetic diagnosis, prenatal testing, as well as carrier detection on a very, very large scale, because typically mitochondrial disorders occur at a rate of around 1 in 2,500 live births to around 5,000 live births. That means there is a very, very large number of people who have suspected mitochondrial disorder. Now, by bringing down the cost, now it's available to a very, very large number of people across the country. Before that, let me stop this last 2 slides of my talk. Let me ask you this question: how many of you would do their whole genomes if it is available? The hands up. Not many. If you have no disease, you assume that you don't have it. I have asked this question to a lot of people, right? And I can approximately say this number is around 30% of this room. If you ask this question in US or in Europe, the number of hands would be 1 or 2. Right? The fact is that we are very different, not just in the genetic makeup, but also in the social outlook of looking at genomes, which is very, very important. And we actually did a survey on a large number of educated people, and this is published today in 2014, to the question that if people would do their whole genomes and actually share that information for public good, provided this available for free, vast majority of people, around 87% of people, would agree to doing your whole genome and putting the data out in public domain, right, with anonymity of course. but provided consensus. And stark contrast, the same survey was done and was published in 2010 in Nature. Even if it was for free or if somebody paid you to do that, there's still not 87% of people who will do it. Right? So socially, there's something very fortunate that we have, and I think I'll stop there. But before I stop, what we think is that to implement this in a very, very large scale, which includes prediction, which includes prevention, which includes making diagnosis much more precise, and of course, making this much more personalized. It's not important that we have all the technology, but we need to have participation of people. And this participation of people is not people like you and me, but a lot of patients, a lot of clinicians, and of course, budding scientists working on this. I'll stop there, and before that, there's a wonderful quote. I don't know who made this quote, but of course, if you don't understand anything about the system, probably you just try to read this. I'll stop there and take questions.
Are you comparing your results with exact database? Because allele frequencies can look inflated or deflated in a small sample size. So, and I just saw you just compared your results with 1000 genome and HapMap, which is like pretty old for now. And exact they have pulled up almost 120,000 genomes and released the latest file. So, are you comparing that, and can you see some significant difference between the allele frequencies of your so-called risk alleles?
So comparing exact is as standard procedure today. I didn't put that explicitly out there. But the real problem with exact is exact doesn't have a lot of South Asian genomes, right? The only genomes which are there in exact from South Asia are already part of the South Asian genome. Right? There's nothing more in terms of the aggregation that aggregated from Southeast Asia. So for practically all the purposes for genetic diseases in India, exact doesn't make any sense. And that's the primary reason why we put together around 1,500 genomes and exomes.
That's the reason is you are basically screening for population-based
which is necessary, because unless you have the population background, you're very unlikely that you're going to end up with the right candidate. Right? If you really go back to OMIM, and I'm sure Parul did this experiment some time back, if you take OMIM and the disease-associated variants, and go back and ask this question, are they frequent in the population? Many of them are so frequent that it's impossible to happen. Right? And you cannot have a rare genetic allele with 10% frequency in the population. That doesn't make any sense. So most of these genetic databases, even when you take OMIM or FINVAR, are full of false positive variants which do not cause the disease. Right? They were marked as causing the disease because they are less frequent in a public database like 1000 genome or exact. But if you really go back to our population and see, they are very frequent. Right? So there's a subtle bias in terms of the annotations that you make, unless you have your own internal database.
Very interesting talk. Thank you so much. My question is this: when we talk about personalized medicine, can we think about the patterns of the population? Like, if some group of a population is showing the same pattern, so can we consider that pattern in personalized medicine?
Yeah. So it's a very, very right question and a very apt question. So what she asked is that when you consider personalized medicine, should it be personalized or should it be based on the pattern in the population? For a country like India, I think personalized medicine is still far, far away, because it's not going to be affordable for every one of you to have your own drug. Right? You cannot spend $5 million to make your own drug. But what is going to be much more affordable is to have populations like this, like the Nalban mutation affects around 50,000 people. Right? So you could make a drug for 50,000 people, and that makes it affordable to a large number. So yes, patterns of the variance in subpopulations are very, very important, because that will allow you to assess what is the breadth of the problem. and now be able to allocate resources appropriately. You don't want to allocate a drug discovery program on one individual because it's going to help just one individual. But you would, of course, allocate the same resources which will help 50,000.
So, if there are no questions, then that's the number of questions.
When segregating your population, have you taken account of environment? Because for particular habitat or location, they may affect. Like I belong to agriculture fraternity. In case of agriculture, when we go for genome of particular plant or species, so environment can affect.
So that's why I explicitly put that slide in the sense that there are two kinds of distinct entities that we discuss here. One is a common variant with less penetrance, and the other one is a rare variant with very high penetrance. Yes, sir. Most of what we work on is in this space. Very high penetrance means environmental effects are almost nil, right? Or modest effects, which are pharmacodynamic effects, where there is a modest effect size on the environment. Right? The effect of the adverse drug reactions also depend upon whether you took the drug before, food, or after. There is an effect of environment, but there is also a very large genetic effect. What I didn't discuss is this part of the spectrum, which includes very common diseases like diabetes and cardiovascular disease, obesity, where the genetic effects are very, very less, variant frequencies are very large, and the environmental effects, therefore, are also large.
Okay. So if there are no questions, let us thank this for his wonderful token of our appreciation. So, we have our next speaker, Dr. Ganesh Bagler. I have the pleasure of introducing him to you. Ganesh Bagler is an interdisciplinary researcher working in the area of complex system, computational biology, bioinformatics, in silico drug discovery, and applications of data science to health and medicine. He has trained as a physicist and also as a computational techniques and computational biologist. And he got his PhD from CSIR lab, namely the CCMB, and he had his postdoctoral training from NCBS, namely the National Center for Biological Sciences, and Max Planck Institute for Molecular Genetics. He is credited with the observation of assortative mixing in residue interaction graph models of protein structures, as well as discovery of exceptional contrasting food pairing in Indian cuisine. His research involves questions on the interface of biology, medicine, and computational sciences. He is an avid science communicator, having delivered lectures across the country, including a TEDx talk and keynote addresses. So he is going to talk to us today about network biology and integrative paradigm for modeling biology. So I call upon Ganesh Bagler to present.
Okay, good afternoon. What I expect to do is take you through landscape of network biology. The agenda here is assuming that many of you would not be very well acquainted with the notion of network biology, although I know as people who are working in computational biology and bioinformatics, you are aware of networks in biology. Many of the speakers before this have talked about, for example, the drug target network or even other networks that have been talked about so far. So I'll be taking you initially I would like to give you some motivation about why we would like to do the kind of work that we do in network biology. We'll take you through some of the basics in a very fast manner, and we'll tell you about some of the research that we have been doing in our lab in this area on various topics. Okay, so initially I would like to tell you a story about a biologist who can't fix a radio. In case you have not heard about it, you would find amusing, I hope. And followed by that, I would like to tell you, as I said, the basics and applications in different areas. I'll be telling about what I have done in my research, that is that of working on protein structures and some interesting work with the protein structures using network biology models, as well as diseases, something in drug discovery, and about brain network models. I know it's a wide spectrum. We are talking about various entities, but I would like to impress upon you the utility of an abstract theoretical model in all of these biological systems. So, this is a story of a biologist who ends up finding a radio, a working radio, without knowing how radio functions, and realizes that it gives, it has an interesting property of generating fantastic sounds, and that's what the biologist now would like to explore about. How does it happen, right? Actually, this radio is that of cancer, by the way, but we'll come back to that. So, the mechanisms which are underlying cancer, by now you would have realized with the map that was shown to you earlier about the various metabolic mechanisms, as well as other networks that have seen throughout this day, is that all these mechanisms are intertwined with each other in a very, very subtle manner, creating naturally a very interesting complex network. Okay, we want to understand that, and we would like to compare it with that of a radio. If cell was a radio, if a disease was a radio, and if you want to understand it, how would you do that? Well, this is a critique of what is called as a reductionist approach. And when I say a biologist, I mean you and me. The reason being that people get offended because I come from physics training, so they think that you are a physicist, therefore you are blaming us to be a reductionist, but not really. So any one of us, when you look at a system, which is what Professor Brahmachari referred to as a elephant and blind men, right, where we tend to take a reductionist approach, and such an approach leads you into a very interesting scenario. Right, so a biologist typically would first of all ask DBT for a lot of fun for buying lots of radios and hang those radios on the wall after opening them. After seeing that there is a very subtle interconnected circuitry inside it, and without knowing the electrical engineering and electronics that's going into it, what a biologist would do is to buy a lot of stones and stand at a distance and throw the stones randomly at this radio with the hope that one of those stones will actually affect one of these components, creating a functional phenotype, okay, and something, for example, in which there is a blurry noise or loss of audio or some other loss of function assay, let's call it, and would like to name them based on whatever one is observing. So from such various studies that are happening, it might happen that one of them would report saying that I found a component which makes the sound of the radio noisy, and will call it a seriously important component, and for the sake of simplicity we'll call it SRC. And somebody else would actually find out something else which is more important for the function of the radio and would report it to be, forget about SRC, I have got an RIC, a really important component. And somebody else on the top of it, as it goes, because the paper has to be published, and because as a blind man you're trying to find out which component is the most important, you'll end up finding something called most important component. And to top up on that, somebody else will catch up with it and say that, forget about all three of you, I publish a story in Cell which is found undoubtedly most important component, right? Now, if that reminds you of what is happening in typical reductionist studies that we have been doing, this tells us about this fallacy of looking at a complex interconnected system as a simplistic system and trying to find out a one-to-one relationship between a gene and a disease or a protein or a functional phenotype, right? Well, that's where we stand today with an understanding which was actually given to us long back in this small article written in Cancer Cell by Yuri Lazebnik, who said that we of course need to have a better formal approach of looking at molecular entities, and as opposed to having cartoonistic picture, we need to have a better picture of these entities. Not that today we have it all, but people like us, we are trying to generate data, trying to connect them together in a meaningful manner, in a causal manner, and trying to get a better systems level picture, an integrated picture, with the hope that we should be able to apply more formal methods such as that of an electrical science, by which we should be able to pin down really interesting, important component with which the idea is, apart from modeling these biological entities, including cancer, we would like to control it. We would like to find a way of addressing the problems that we have found there, including pharmacoinformatics, where we are trying to find drugs which are specific for a certain population, let us say. This is a known picture to most of us sitting here who are acquainted with the fundamental understanding of biology, right? And this tells us about the logical flow of information that we have known to be there, right? The central dogma from DNA to RNA to protein. And what we now know today is that probably it needs to be extended, enhanced by having a systems picture, right? And this is where network biology pitches in with the graph theoretical aspect. So, what are these complex systems? Because I take pride in saying that I work on complex systems, right? It could be from biology, it could be from social sciences, or it could be from technological sciences, but these are all complex systems, and these are something which are made up of large number of sophisticated functional elements, each gene, protein, for example, and they are intricately connected, linked with each other to perform specific tasks, specific metabolic pathways, specific functions in the cell, and these functions cannot be otherwise be performed by the small subset of it. So that's a generic definition that I have cooked up to give a sense of what these complex systems are. Most importantly, these complex systems, apart from having a networked connections, they also have nonlinear processes which are ruling there, and further giving rise to what could be called as an emergent phenomena, right? Emergent phenomena that is coming because you know that each of the entity inside a cell is that of a chemical, largely a non-living entity with some specific function, but when it is coming together it gives rise to a living entity which is reproducing, living, etc. Right? Okay, so that's the emergent phenomena that we are talking about. So today, as I said, I'll be taking you through initially the networks, basic network concepts which some of you might be aware, but bear with me, and we'll be talking about these radios, complex systems that we are looking at. And the radios that I'll be raising today will be that of a protein structure, such a small thing at the nanoscale. How can you look at it as a network? Further, you can talk about a cell, which is a diseased entity, let us say, how to model the mechanisms of a cell, molecular interactions of a disease, let us say. Okay, the drug discovery process itself can be seen from the prism of network science and probably can come up with some answers. And finally, I'll take you to the most interesting probably entity that is with us, which is an example of complex system, that is that of a brain. Okay, I'll be taking you through this. All these studies are something which we have done in our lab, and not to say that we have done anything great in that, but of course we have got forays into each of these areas with some interesting insights in these systems. So look at this one. When I'm looking at these radios or complex systems, I'll be looking at a scale of nanometer, the protein structures, going up all the way up to micrometer, where a cellular level network is performed, to the level of centimeters, where the brain networks are there, including that of primitive organisms, and finally going to society. Although I wouldn't be touching upon this part, I'll be talking about mainly the bottom layers. Okay. So the graph theory, I mean, I would also like to impress upon you that while many of us would be highly interested in finding problems or prevalent solutions which are already there, including societal problems, etc., probably there is some sense in working on some of the problems without knowing there is a utility for it naturally, right? One of the examples is that of graph theory. If I'm standing here today talking about some of the applications of graph theory in biology, it was because of this particular city called Königsberg. Okay, and it's a century-old story. So I'm winding up in 40 minutes. So it's a big challenge. So there was a river that was flowing through this Königsberg, and these were the bridges that were present in that, right? And in a leisurely manner, the citizens, people living in this village, asked a question while taking a morning walk about, could it be possible for us to walk through all the bridges once and never cross the same bridge twice again until reach to the same point back again? Start from, let's say, A, go through all the bridges such that you cross every bridge only once and return to the same place again. That was a very simple question, and you might think it's a question asked by a lazy mind not having any work to do. Okay, but turns out that this question was very tough to answer in a rigorous manner until Euler came up with this idea, abstract notion of graphs, and converted this problem into a corresponding graph, and he finally answered it with a theorem. Okay, in this question, for example, he said that it's impossible to have such a path, circuit in fact, to be able to reach back to the same point again, because there is a precondition that every node must have even number of connections. Okay, that was the theorem. But now, what started with a very simple situation like this, which I told you about, turns out that this can actually lead to interesting theories, which can be further used for many of the realistic situations. Okay, some of the concepts that I would like to take you through. One is that of paths. when you have such networks, including transportation networks, one of the simple questions that we would like to ask is about what are the paths available here. You must have heard about traveling salesman problem probably, finding out an optimal path to reach from one point to another point, for example, and there are similar such questions which can be asked, wherein one is asking about what is the shortest path from a point to another point, and there are, in the presence of multiple paths, we want to find out what's the most optimal or the shortest path. Okay. The other question, other definition which is raised in the area of networks is that of clustering. Okay, I'm trying to take you through a history of roughly last 12 years or 15 years roughly, in which some interesting work has happened in the area of network science. The clustering actually refers to if a node is connected to 4 other nodes, where I'm referring to each of the entity as a node, how many of these neighbors of this particular original node are interconnected with each other? How many of my friends are friends themselves, creating a triangle with me in the center? That's a question one is asking, and this refers to what we call as a clustering. How well clustered is the graph? In the context of this, one of the early study that was done was that of by Watts and Strogatz. From again a very perspective of a dynamical system, they looked at a network as an entity which is made up of large number of nodes, but connected in a very regular manner, very nicely connected. Okay, and they proposed that if you start with such a graph, which is a regular graph, and start increasing randomness in it by rewiring the edges in it without changing the number of nodes or edges, but simply rewiring them, you will go through a spectrum of various conformations, network conformations, to end up into a random graph. You can imagine that. If I were to rewire each and every edge, you will end up getting a graph which is completely different from this and a random one. Okay? The interesting observation they could do from here while comparing with the real world graphs was that in this spectrum, in somewhere in between, you end up getting properties which are very interesting. What are those? When you look at characteristic path length, the average of all the path length, shortest path lengths that I told you about, it starts from where it is very high in a regular graph, because you need to pass every other city or node to reach to some other point, right, in between. So it will be very high. Similarly, the clustering is also high because it's very nicely triangulated graph. But when you keep on increasing randomness or rewiring, the clustering coefficient falls in a very slow manner, whereas the characteristic path length falls fast, creating a central region which is very, very favorable because it has got very short path length. It's very easy to commute on such a graph from one node to another node, and at the same time it is very nicely clustered as opposed to random graph which is not so clustered. Okay, this is what was referred to as small world. Okay, many of us are aware of it, about how we live in a small world by meeting an acquaintance who is a friend of a friend of a friend, and thereby declaring that it's a small world after all, right? So loosely speaking, that's the concept that we are talking about here. Okay, high clustering, in addition to having a very short path length, seems to be a very interesting property which seems to be characterizing most of the real networks that are studied. Technological networks, transportation network, social networks, as well as, to add to it, biological networks. The other property that seems to be interesting is that of the distribution of connectivity of the nodes, the degree distribution. Turns out that if any given graph, including biological, if it was completely randomly connected with each other, with same number of, let's say, proteins and protein-protein interactions, or same number of metabolites and metabolic reactions which are connecting these. If it was so, then the degree distribution would be something like this, a Poisson distribution with a very thin tail falling on both the sides. On the contrary, most of the real networks, it has been discovered in the last 16 years or so, they are characterized with what is called as a power law or a scale-free distribution, suggesting that most of the large number of nodes tend to have very low connectivity, low degree, like shown in this graph, okay, and a small number of them tend to have exceptionally high degree, high connectivity, indicating that there is a presence of a few nodes which are heavily connected, rich, and that's why they are called hubs. Okay, this is a characteristic feature, and very interestingly, this feature seems to be present even in biological networks. When you start looking at protein-protein interaction networks, you will end up getting a few proteins which are heavily connected, and therefore they are kind of the backbone of the whole protein-protein interactome. They are the ones who are stabilizing most of these PPIs, the protein-protein interactions, and that is true for many other networks as well. So this is the other property, and interestingly, this property was shown can be pinned down to the lethality in the case of yeast protein-protein interaction network. In fact, this was one of the earliest study published in 2001, which was kind of very famous for analysis, system-level analysis of protein-protein interactome and connecting the topological property, that of a scale freeness and the presence of the hubs, with that of lethality. More central a protein is, mutation in it will lead to lethality of yeast. That was the observation that was made. Although the correlation is not fantastic, there was an indication of the fact that, which is what Professor Brahmachari's talks about, that the hubs are the Achilles heel of this network. That while the network is extremely robust under random circumstances, random mutations, random changes, that is what you are expecting in a natural circumstance, when these hubs are targetedly attacked, like in the case of a biotechnology lab, where we are trying to mutate the hubs, that's the Achilles heel. You know the story of Achilles heel, right? Achilles, who was extremely powerful under normal circumstances, had a weakness. When hit upon a certain part, he will be killed easily. That was the idea, healing, right? That's why it is called so, right? So this was another idea of a network concept, may I say, graph theoretical concept, which seems to have very relevant functional connection. Well, going further, beyond looking at the small worldness based on the clustering and the scale-free architecture indicating the presence of the hubs, other models were also created suggesting modularity and hierarchy in the networks in general. Most of the networks that we see around tend to have real-world networks. They tend to have modules present in them, which are interconnected to create a large network, and some kind of hierarchy also. And by looking at metabolic pathways network, again in one of the early pioneering studies, it was shown that in the case of E. coli, these metabolic pathways could be clustered using such kind of a hierarchical clustering, and the modules that are present in these metabolic network, they can be associated with specific pathways like lipid synthesis, carbohydrates generation, or amino acid synthesis, etc. Okay, that was another interesting thing. Now this is what is telling us that, now notice, while we made joke of reductionist approach, we are using the data coming out of the reductionist approach. So the joke was not on to say that everything that is done in the reductionist approach is wrong. It is to say that it is good to know that we are blind to the larger picture while you are looking at an elephant and trying to find a smaller picture out of it, because you are limited right now, right? The data that was collected from small experiments were pulled together to create a system biological picture, a network picture, and further analysis was applied to find out modules which can be obtained now using computational techniques, which seem to be very nicely correlated with biological functions. So that was a very nice utility coming out of a simple question that was asked in Königsberg, saying that, can I reach to the same place back if I were to travel through all the bridges only once? Okay, so I hope you appreciate the usefulness of the useless knowledge, right, that is coming out of it, without having to every time defend ourselves by looking at a specific problem per se, while it is inspiring in itself. Further, beyond the modularity and other properties, one of the things that has been talked about in the biological networks is, are there simpler entities or circuits, let's call them motifs, which are the building blocks of these larger networks, all type of networks, including biological. Okay, such as the pattern of connectivity that we see here, some of them, let's say talk about this one, A connects to B, B connects to C, and C connects to A back again. What kind of a network would this be? A connects to B, B connects to C, and C connects to back A again. What would you call it? It's a feedback, right? It's a feedback loop. In a similar manner, you could have other kind of mechanisms that are present in a 3-node motif, similarly for 4-node and 5-node motifs, right? And this particular study, for the first time, investigated what kind of such structures or subgraphs, as we call them, are present in a larger number in many of these biological networks or even other networks, and came up with a realization that some of these subgraphs seem to be predominantly enriched in these networks that we are looking at, presumably because they are functionally relevant. Okay, now the study of these motifs has gone a long way now, trying to pin down specific mechanisms in which these motifs are contributing to, depending on what kind of network you are looking at, and of course biology is one of the hot areas in which you can look at. One of the areas that, these were the studies that I'm talking about for last 10 years or so, 10 years back or so, one of the studies that has come up very recently in the last 5 years or so is that of controllability of the graphs. Notice that beyond looking at existing networks, trying to model them, trying to understand them, other thing that we would like to do is to control them, okay, to be able to have a way of pinning them down, right? Controllability analysis, which is borrowed from electrical engineering or control systems, tries to borrow this idea, notion and the theory with an approach trying to find out the nodes which are most important for controlling the dynamics of the system. Okay, notice that so far, largely speaking, I have not been referring to the dynamics part of it. That each of the gene, each of the protein is being synthesized on a continuous basis and the relationship between them might be changing. The concentration of molecules might be changing, right? It's a dynamical system as opposed to a static picture that I was trying to portray, which is far from reality. In such a dynamical system, and if you can have a good data available for that, if you could find out which are the points of interventions by which you can control the dynamics of the system. Imagine that being the brain system, imagine, right? And you push the brain from a depressive state to that of a pleasant state, for example. Or you talk about a gene regulatory system where you would like to push the cells back from cancerous state to non-cancerous state. Right now this seems on a very abstract level because we have not found very precise ways of actually controlling them. But this kind of a theoretical analysis in the area of networks has become very popular these days and probably can be applied in various areas. I would like to show you some examples. So I gave you a summary of what has been happening in the area of complex networks, network theory, network science for some time. Of course, I missed out a lot of things. Advances that have happened in the area of algorithmics, trying to find out innovative ways of clustering the data, finding out the modules in the biological systems. One of the simplest one I showed you, which was older one, but now there are innovations that are happening in that. There are different parameters which are being defined in the context of a specific data, which can make sense in the context of a radio that you're talking about. If it was a gene regulatory network of a cancer, what kind of a methodology would work better as opposed to if you're looking at a brain data, for example. Okay? Well, all of this can be loosely summarized in terms of this picture, which was given some time back but still valid, is that networks tend to have a large-scale organization where you can say the whole network is a small world or a scale-free. They could have functional modules, a module which is responsible for lipid biosynthesis versus that of amino acid biosynthesis, etc. And there could be something like pathways or motifs which are functionally critical for a given system, but stand at a level which is more and more specific to organism. Okay? And finally, something very, very specific to individual organism is that of the genes, the mRNA sequences, protein structures, and the metabolites that they tend to synthesize. Okay? So that's the overall picture we have when it comes to biological networks. When we work in a lab, complex systems laboratory, we have been looking at these entities from the perspective of modeling, controlling, which is what I told you about, and finally, why not designing? Can we have bio-inspired systems which have learned design principles from these evolutionary entities which have survived over a period of a time? Could it be possible for us to learn something from this system so that we can learn about robustness from the biological systems under circumstances? Notice that unless it is inserted with a heavy problem or an insult, most of the biological systems have evolved over a period of time to survive under natural variations or stresses that they come across, whether it is a plant or an animal, right? Can we learn these principles, the architectural principles from that? That was the idea, and trying to apply it in the area of biology and medicine is what we are trying to do. So let me take you through the first example. Protein structure. What do you think a node would be here in a protein structure? If I were to force upon the abstract graph theoretical model on a protein structure, what would be a node? Discrete entities, what are they? One would be secondary structures, what else? Secondary structure as a unit, what else? Something smaller than that, probably. Amino acid, for example, right? Amino acids seem more like a building blocks of the protein chain, okay? One of the simplest model that can be, by the way, when I tell about this research, I would like to also tell you about the fallacies of using graph theoretical modeling in a wrong manner. Meaning, notice that while you can construct the abstract graph theoretical representation of anything typically present, which has got abstract discrete entities which are interconnected with each other, the kind of question that you are asking based on these systems is the most important thing here. Depending on the question that you want to ask, whether the abstract model, whether it is relevant or not, completely depends. You might end up coming up with some kind of parameters which are absolutely useless in the context. So let me tell you about that also. So here, in our case, we are using amino acids as nodes, and we are simply using a spatial model wherein any two amino acids which are close to each other in the three-dimensional space because of hydrophobic interactions and other interactions which are responsible for the folding of the protein are the contacts between them. When you do that, you will end up getting a contact map like shown here for a simple protein 2-PDD, wherein you have a backbone connection between node number 1, 2, 2, 3, 3, 4, etc. followed by other interactions which are happening in such a manner that these are representative of the alpha helices in which the nearby amino acids are interconnected with each other, and you have also long range connections without which the protein cannot have its globular tertiary structure, right? So such a map can be created, and I am trying to deliberately point it to you that remember the Watson-Stogas experiment that I told you about? I am trying to create a replica of that wherein you have a completely regular kind of a graph. You cannot expect a protein contact map of this sort which has evolutionarily evolved, like it has evolved over a period of time. It would be rather too simplistic to have such kind of a contact map. You could also not have something of that sort which is completely randomized and it is not realizable in the three-dimensional space, whereas something like this seems to be more realistic. So we built maps of these networks, and we found, not for the first time, that these are indeed small world networks, have high clustering. You should remember that, the neighbor being connected with each other kind of a property, and also have a short path length. Beyond that, what we also wanted to find out, what are the new features, if it all it has got, and what are its functional relevance. And that's where I ended up discovering what is called as assortative mixing, in which a network, it's a kind of a network in which hubs are predominantly interconnected with each other. It's a rich kind of a connectivity, okay, that you find in a network as opposed to rich-poor connectivity. Okay, this was something which was unusual because most of the networks were known to have this assortative mixing, whereas a rich node is connected to large number of poor nodes, isolated nodes. That was the kind of a pattern which was found there. Well, we found that this assortative mixing, incidentally, when you study simple single domain two-state folding proteins for which the rate of folding is available experimentally, if you look at the assortativity coefficient and the rate of folding, we found that there is a fantastic relationship between them. The more assortative is a protein, more the tendency of rich node amino acid connecting to other rich amino acid, you tend to have better rate of folding. Okay, that's what we found, but this one is a negative correlation with another parameter called clustering coefficient. Okay, very interestingly, what it tells us is that while the folding has already happened and we are looking at a native state structure, by looking at the native state structure and constructing a graph theoretical model based on that, using that static picture and the network properties, you are able to derive something interesting about that protein, which is that of the rate at which it has folded, whether it has folded at a microsecond speed, very fast, or at a few seconds. That's what you are able to derive. That's a very interesting thing that you can get out of it. But we realized that this model that we used cannot, for example, distinguish between thermophilic and mesophilic proteins. That is because the details are too subtle to be caught with the help of amino acid contact based network. Well, that's a fallacy. Remember that? If I were to try asking that question about what are the topological differences, network based differences between the two, I would not be able to find it. In fact, we wasted time in doing that. So that is where I would like to point out the limitations of such models, network models. We need to know that. Well, in terms of application, what we could do is that using the same graph theoretical model for a superoxidase dismutase from Potentilla astrosanguinea, a Himalayan plant, in IIT in collaboration with the experimental laboratories there, we could suggest few mutations which could be used for making this protein thermostable. In fact, it ended up becoming hyperthermostable. Okay, so that's a utility I would like to show you. To summarize, at the nano level biological systems such as protein structure, we could use graph theoretical models when appropriate relevant questions were asked, that of I just mentioned to you, and we could actually end up with an application coming out of it, not to mention a fundamental discovery about assortativity being positively correlated with the rate of folding. The next one that I would like to tell you about is about diseases. Okay, we already heard enough of that. In fact, these are very relatively simple models compared to what you have heard today, wherein now you are having more and more specific data. In fact, patient-specific data can become available. we are looking at general picture of how diseases are interrelated with each other in terms of the genetic mechanism that they share or don't share. Okay, quite clearly, this is a complex picture, quite clearly telling us about why the the radio model was wrong, the reductionist approach was wrong. It was impossible for us to find a single gene specifically connected to a specific disease because it's pretty interconnected. Okay, and we need to take into consideration details, data at different hierarchies, which is what is still happening. Okay, we need to look at the data at the metabolic network level, at the level of disease level, and for that matter, even at the level of societal connections. Okay, we need to find out these information, collect a lot of data, which is what Professor Brahmachandri was also emphasizing upon, to be able to build better systems level models where, incidentally, graph theory will play a big role, because that is a very, it's a theory which has developed over centuries and have interesting properties which are available, lemmas and theorems which are available, which can be exploited for asking specific questions on biological data to get answers, interesting answers out of it. So in our lab, we had studied various diseases such as cancer, diabetes and asthma. The idea was to look at the etiological basis of what are the genes, proteins which are specific to the disease, model them as network and find a way of controlling them by identifying targets of these diseases. This is not to say that this work has not gone into the experimental assays as of now, but we hope that some of the studies that we have done can be taken into experimental verification. This is a broader picture I am presenting you, wherein network biology was applied to construct network models and find out targets, and at the same time, very interestingly here, we were trying to bridge data which is available from traditional literature, okay, such as Ayurvedic literature, but has been confirmed with modern studies, available from that, like plant extract efficacy, so as to build databases of plant-derived molecules and use them together with the help of virtual screening and molecular dynamics to predict molecules which could be potentially efficacious against a particular target, specific target. So, well, that's what we did, and we also connected some features that are food-related feature, but without going into the details of that, I would like to say that one specific study, here we have predicted molecules against aldose reductase, which is a potent target of diabetes, in a rigorous manner, and is kind of very well appreciated because we started with this idea, notion of having a systems picture, at the same time having molecular data of plants and information coming from traditional knowledge, which can be further used in addition with, along with the computational techniques. The next thing that I would like to talk about is, again, application of graph theory in the context of another context, beyond diseases. This is that of drug discovery. Again, this point has been elaborated upon to identify a specific problem, that we need to find computational strategies to shortlist the drugs, candidate molecules, which can be further taken into the drug discovery pipeline. So what we are trying to do is to use a picture which is shown here, that the drugs, because of their off-target interactions, non-specific interactions, end up giving us effects, reactions which we call as side effects, adverse reactions. And the data of this, once it is available, which is, we are using right now the open source data that is available from Drug Bank and CIDR to construct a tripartite network of drugs, their associated targets, and the same drugs and their associated side effects. Once you create such a network model, a lot of interesting things can happen. This is another study which my student Rakesh Kanji has been working on, completing his PhD on this, where after building a pipeline for merging and integrating such data in a flawless manner, so as to have final integrated data of drug target and side effects, the idea is to find out what are the different ways by which you can more and more accurately predict the side effects a priori. So that if you know that some of the side effects are not so, something which is toxic, you rather not want to take that particular drug for the drug discovery pipeline. And also look at how the drug is interacting with various genes in the context of the what we call as a genomic space, by which, again, using graph theoretical features, we would like to find out what kind of graph theoretical features are more relevant towards making this prediction. So we have looked at various features like degree and other spectral properties to find out which specific properties are more relevant towards prediction of side effects. So this was another, by the way, did you notice that I am telling you about examples of network models? So this was another example where we had a tripartite network model which can be used with a specific question. How do I build mathematical models with which I can predict side effects more accurately? And that's where we are trying to use the empirical data. finally, I'll talk about the most philosophical question, which is that of brain. Okay, and about how the brain functions, what are its properties. And that is where I am kind of rather enchanted by this view of Francis Crick, who says that it's a very, again, a very, may I say, atheistic, spiritual view of the brain, which says that you, your joys and your sorrows, your memories and your ambitions, etc., are all, including free will, are all in fact no more than the behavior of a vast assembly of nerve cells and associated molecules. Okay, many of us who believe in souls and spirit would not want to take this. I know that, right? But nonetheless, question is, can we ask scientific questions, with the data that is available? That is where another student of mine, Rahul, who is there in the audience, has asked these questions for last 3.5 years, close to 4 years now, at different levels, looking at the neuronal connectivity of the, in the case of C. elegans. We have not touched upon the human brain yet because we do not have comprehensive data, C. elegans being one of the simplest organisms. Further, at the level of neurons and synapses, the molecular molecular details are available, and going all the way down to the genomic scale. Okay, in the case of C. elegans, specifically we have been probing down the question of controllability. But in general, this particular area is open for at least two types of various philosophical questions. One, we should not say there are philosophical questions. These are questions which are scientific and data-based, but these are two different school of thoughts. One is to look at the anatomical connections and construct a structural brain network to further analyze the graph network to see the connection with the function. Second is look at the functional data. It could be coming from EEG studies or it could be coming from fMRI. Further, to pin down the correlates or markers of specific diseases which are known to be located in the brain. Okay, so in the case of C. elegans, we have constructed the C. elegans neuronal network to look at the connectivity pattern based on the distance between all neurons, and we have observed there to be something called a distance constraint. There is a power law here, and using this idea, we have constructed models which we have published as a distance constraint model, synaptic plasticity model of C. elegans. This is a network model. Very interestingly, controllability theory allows us to identify the driver neurons. Neurons could be critical for the for driving the dynamics of this particular organism, and which we have already pinned down to the level of genes and the phenotype. Another paper published on that. And this one is a more of physics-oriented study where we showed that using a distance-constrained plasticity model, we are able to predict specifically with an accuracy of around 77% at a certain parameter value, distance-constraint value, the neuronal structure which could lead to controllability architecture similar to that of the C. elegans. Well, that's an interesting insight that we are getting into the brain networks. And finally, the study which was done with another student at IIT Jodhpur, Megha Singh and Rahul. In this study, we have investigated the brain functional networks of subjects coming, schizophrenia patients as well as healthy subjects, compare them by constructing their brain network models, the way I told you, and finally pinning down on graph theoretical properties which are shown here, which are best discriminator between the two states. Okay? Well, in summary, what I would like to tell you is that the brain network seems to be one of the interesting paradigm in which a lot of graph theoretical analysis can be done, and the data is becoming more and more available. What I showed you was one of the simplest models, that of C. elegans neuronal connectivity, and a very simple data of schizophrenia patients of functional brain networks data. But this can be used further. If you have the tools available, such as that of graph theory, and if you know what works and what doesn't work in the context of brain networks, we have lots of data that is going to become available, wherein we can ask more interesting questions, trying to pin down the model as well as the control of these networks. So I would like to summarize that we looked at protein structures, we looked at diseases, trying to identify the targets, which was again shown in the case of Mycobacterium tuberculosis. That was the Achilles heels, the hubs, or betweenness nodes, which were found to be more relevant in that case. Further, we looked at the example of drug discovery and how data available of drugs, targets and side effects can be modeled as a graph to ask relevant questions, both for predicting side effects as well as for using them for repurposing. So the strategies can be developed out of such data, network model strategies for repurposing of some of the drugs. And finally, the brain networks, where we seem to be still scratching the surface because of the lack of the data. Well, this is the lab website. You can look at more work. For the paucity of time, I'm not going to talk about some of the work which has become more popular, that of food, Indian cuisine data, which we have looked at from a systems perspective and come up with some of the interesting observations about the Indian cuisine and the molecular patterns in that. Those of you who are interested in network biology would like to learn, and I know that not everything can be learned in 40 minutes. There is a course which will be running in the monsoon, that is network biology course, and this course will be open for all of you, available from this backpack web page, and you are most welcome to join the course, follow the lectures, and if you wish to do projects, which are projects are part of this course, by the way, although you wouldn't be crediting for it, but in case you are interested on working on projects, do email me, I'll be available for you. So this is the network biology course which you may want to join. So in summary, a person like me who comes from physics training and a computational training, what I have learned from biological system is that these amazing systems of various types can teach us something interesting about how networks are created, what kind of architectures are present in them, and probably can help us in designing the future system. So while doing this, we have asked questions which are knowledge-based, hypothesis-driven questions. We have constructed databases which I did not talk to you about, medicinal plant molecular databases which are all available, as well as we have come up with applications including an Android app called Flavidoscope for generating novel recipes. All this comes from asking a question from a complex system perspective and knowing that we should not be doing the same mistake that a traditional biologist did when he or she looked at a radio. That you don't look at it as a reductionist system. If you look at it as a holistic system, probably we have a hope for finding good answers from this system, learning from these systems. I'd like to thank a lot of students who have contributed to this, many of them from IIT Jodhpur, the PhD students, Rakesh and Rahul, whose work I presented, Rakesh's work I did not present, Shivalika's work I presented, she completed her PhD recently, and many other students who have contributed, both BTech and MTech students, including those from IIT Delhi, who are sitting in the audience. So thank you, thanks a lot.
Questions, please.
Any specific questions? In any case, as I said, I will be available in the tea break, but otherwise also feel free to shoot a question and email to me.
So, if you do not have any questions, you can ask him. If you have any questions, you can ask him during the
Students don't ask these questions in front of faculty. I don't know.
I don't know. Probably.
But if the faculties go away, I have seen that question. But I have never understood.
Actually, they ask questions. I have to, you two people came. I think so that you are actually the people that who are not facing this question. Okay, if there are no questions, let us thank this speaker and open up our presentation. So we'll break for the tea and we'll come back to the you are following me.
Welcome to the last session of today's symposium. I have the pleasure to introduce you to eminent speakers for the last final session. The first speaker is Dr. Dinesh Kumar, who is currently working as principal scientist in the Center for Agricultural Bioinformatics, ISRI, Pune. Earlier, he worked as a senior scientist at ICAR NVATR kernel. He obtained his PhD in biotechnology from BHU and he did his postdoctoral fellowship from Iowa State University. Interestingly, he also has a LLB with specialization in patent and cyber law. He also has a postgraduate diploma in technology management in agriculture with a gold medal from Central University, Hyderabad. He has published in top journals like Science and PNAS. His specific efforts has been towards developing web servers for identification of domestic animal species, and he had led his team to develop probably the world's first such web server, which used more than 50,000 DNA markers to classify goats using technology like patient classification. He has later extended his techniques for identification of crops also. He is currently involved in genomic data analysis of crops, animals, fishes, microbes, insects, all these kind of things related to agriculture, and that's probably what we're going to talk here. With this, I'll leave the podium to Dr. Dinesh.
Thank you. Respected all the participants and our Guru, Professor G.P.S. Raghu, sir. And the introduction itself, which was referred like analysis of 50,000 fingerprint for identifying the breed. In fact, when we have been struggling, then just like some Kabir, you get some right to do out of the system. So we met Professor Raghu, sir, and he was the key person. And today I am so happy that as one of his disciples, or the F1 generation we call it in agriculture, that we had those academic genes in very short interaction, but it expressed, and we got few publications in leadership of scientific leadership of Honorable Professor Raghu, sir. It's not inflated, it's reality, you will see. And very interestingly, probably he may not be knowing his own importance. It happens with all exceptionally born genius people, not acquired genius people, like Professor Raghu, sir, that his concept is now moving towards the real protection of intellectual property in form of the variety. The server we developed for animal doesn't have the intellectual property right like variety, but variety is having, and we are carrying forward with his blessings and the insight of the academic genes. And he is not expecting any kind of authorship or any kind of, I will say, appreciation. Even nobody has thanked him from my organization for mentoring our group of scientists. So that defines actually Professor Raghu, sir. And when I got the invitation, I came here at 9:27 just for his respect, because I understand if he goes to moon and if he calls, I will try my best to reach there. Because that kind of spirit we have, he may not be knowing that how much respect he is enjoying, but he is never looking forward. He is an exceptionally different person. So with this, perhaps it is the greatest ever pleasure of my life to give a little reflection back what we learned from him and how we are going to use. So the turning point in the life of biologist was like working 15 years with your white coat and your apron and your micro pipette. In 2007 to 2008, 1 year, exactly 365 days, something happened. And I was having the opportunity to work in a cow genome project which was computationally intensive in terms of assembly and the annotation, and also a wet lab work where heavy computational intensity is required to locate one gene and its mechanical connection causing dwarfism related to the wheat industry in the US. So that actually transformed me, and then we started searching, and we found that Professor Raghu sir is in India, is an exceptional person, and we wanted to become 10% of him by acquiring something till we get retired. In that spirit, I changed my biological lab and became first biologist of the Statistical Institute where supercomputer was being developed. So I will be speaking about my center, then the real national goal in terms of demand gap and researchable issue, like I am the first biologist not to solve any problem, but to create problems to be solved, or a problem creating person within the system. So the first part is, can we have really genomic solution of agricultural problem? If yes, can we intervene computationally? If yes, then how? And where is the proof of concept? Can we really move forward? So I will be taking just brief outlines which country is preparing now to move towards the genomic solution steps. And then briefly we will have a quick look on domestic animal, how far the world has gone in computational genomics, and how far crop and how far fish. And it's not just copying music and producing here like your Bhakti Larry, but it has to be actually real in the sense of existing Indian problem. So that outlines we will see, and lastly we will see how computational genomics is going to be used in germplasm management, especially India is rich of genetic wealth, and the kind of piracy Bioinformatics has opened, that now without any germplasm movement, piracy is possible. Piracy is possible of germplasm by sending the DNA sequence, and lot of things are happening. And lastly, I will conclude, I am sure, within 40 minutes. And I won't bore you, I am sure I won't make you to feel sleep, because many points are so exciting which I will be sharing. Now, only one line, that we have a very big, huge facility. It is of 34 crores, it is in Delhi, and it is the Ashoka High Performance Computing System with 70 teraflop speed. And if you see the center, precisely go to its ranking, which is done by Indian Institute of Science, Bangalore, you will find in top 30 we have 2 computational systems. And Professor Lavosan has visited, and we dream that how best we can use in collaborative mode, especially in public funded institute like this center, in our center, utilizing this facility to solving some common goals of mutual interest that we look forward. Now, coming to a very traditional picture, that though I have never studied agriculture, but I feel pride to learn agriculture from any agriculture scientist we met. So in last 2 decades, by interacting with agriculture scientists, we learned that per unit area per year, your yield in terms of kilogram is less than 2%. That means it is too slow to improve. So certainly we need to look for some alternative. And these alternatives are, you go for sequencing, and you go for sequencing of every varieties or breed and generate the SMP, then go for SMP, check after applying the filter, and go for precision phenotyping, go for association, and you start experimenting. So in one line I have finished the entire thing, like what is the proof of concept or the workflow you can visualize in using the genomics in agriculture. But speaking is very easy, but in real terms there are several issues. Slightly depth, if you go from hand lens to compound lens, you will see that we are sequencing, then looking for its variant, maybe your length polymorphism or structural polymorphism like SNP or SSR. Then high throughput genotyping, because we have a huge population and that kind of analysis. So definitely digital tsunami will be there, both in terms of genomics data and the genomics data. And if you are starting for selection, it requires computational intensive work, and it fully justifies that computational biology is required not only in human, but even to the germplasm, which gives the human food, that is also requires that kind of computational intervention. Now a very peculiar situation is India is rich or poor. It's very difficult to answer. But if you see that 20% of the population, they have enough money, but they can't digest all kind of food, because doctor says they have prescribed that this is you are diabetic, this is you cannot. And at the same time, 20% of our people cannot afford the food, maybe because of the price or the bioavailability, whatever the reason may be. That means India is a very unique country where you find class difference in 2 extremes. And here, can we imagine that in this dichotomous situation, computational genomics in agriculture can do something? I apologize here to show this picture because this is really frustrating and humiliating, but I am just quoting Times of India, and this is the 2016 report of the hunger. If you see India, it still lies at the place where we feel really uncomfortable. And if you see how far we are better than your neighboring countries,
then except Pakistan, all our neighboring countries, they have much more secure in terms of food security, in terms of malnutrition and other things. All the details are there. So I am trying to give you a picture that though we have surplus food, that's reality, but at the same time, world's largest malnourished people still lies in our country. And the goal of 2030 that everyone will be hunger free, the way we are increasing, it's really questionable unless we have one approach like computational genomics. It may not solve, but this is one core apart from the other several approach. So, if you see the food security in terms of the disease, then definitely India is going to be, or it's already a diabetic capital, and by 2030 we are going to have huge number of diabetic people in this country. Why I am talking about this? Because if you see the Sarvati, the Madhya Pradesh chapati or the wheat you get, it is more than the double price of governments where minimum support price is still people buy it. It has a lot of things related to that. If you see the your recently released two rice varieties, they are lower glycemic index. So now the time has come. It's not only the organic food, it is also the safe food where genomics can play a very important role. And I am not going to the detail of the cardiac disease and its figure, but if you see the total demand in the agriculture, it is increasing. Commodity wise, it is increasing, but this one slide makes it very simple to understand that we need to increase fourfold productivity. The way urbanization is increasing, you can see even this area also, like all were agricultural lands, but now because of the change in land use pattern, now you can see urbanization. So the land is actually decreasing and population is increasing. We are going to be 9.2 billion population, and our wheat itself is we require by 2070 we require 60% more wheat globally. And if you see the temperature, it is rising. 1 degree rise in temperature is going to affect 6% of the wheat productivity, and we expect 29% of the wheat loss because of the climatic change, which may be up to 0.5 degrees Celsius depending upon Rabi or Kharif season. So the take-home message from all this situation I am trying to give you is, so even if you are safe, but you are not safe, the way you see at global level, at global population, or global market, or global challenges in terms of the resilience. Now, looking at all this, can we really, let us imagine that for 1 minute we believe that Dinesh Kumar is saying computational genomics can solve agriculture problem, but don't trust on me. I may make claim to everyone to inflate my CV and get some money from government, but in reality it may not work. That's the logic definitely one should feel. But at the same time, you would like to know that forget India. Let us see really what has been done, not just to copy, but to have some confidence in the technology which is not existing in India, but maybe existing some part of the world. And let us see how far it is relevant. Should we really import it, or should we translate it, should we transfer those technology to our problem? Then only we will be able to understand without being biased. So I will be taking very quick example of domestic animal, fish and crop, like how far the world has gone using agricultural computational genomics to solve some of the agricultural problems. I am not saying India is going to do this or we are going to do this, but at least see the concept part, then we will see how far we are going to see. In one slide, if you ask that agriculture is domestic animal and your fish and of course your crop, then in one slide let us see how many genomes of agriculture, especially of domestic animals, they are sequenced, how many of them are done by India. India failed disastrously and we failed disastrously. Then we could not even completely sequence even one protein. Now coming to, once you sequence the genome, then different varieties or different breeds you need to sequence, you need to mine the SNP from respective gene pool, you apply the filter, and you make at equal space over the chromosome, you have the SNP, and once you have the SNP, you can use it for association study to accelerate on the desirable allele or genetic gate. That is in nutshell in the very first slide we discussed. Now coming to, in one slide, if you see the assembly, except Mithun, it is available for all, and if you see the SNP chip, except YAP and Mithun, it is available for all, but if you see how many of them India has developed, it's a big question, because we have not. Now, still let us have a lot of doubt that milk yield increase, protein yield increase, because we buy the milk by different colors looking at the polybag. and we call it like 2%, 3%, 5%, 7%, and that's very easy to understand, 5%. But if you see the rate of genetic gain, if you do by classical breeding, and if you apply the genomics, there is a vast difference on the published literature, and those are not from India. Now, if you see here, in this graph particularly, if you see this kind of association study, if your sample size is 2000 or your sample size is 10,000, your predictability becomes better with the larger size of the samples in this kind of genomics study. In India, you won't find, other than your NTTP and private NGO, I cannot make any comment over there, but in the national herd, you won't find even 1,000 animals along with the DNA samples and the phenotype. So the technology may be good for some other country, but in our country, it's very, very difficult. And these are the data which shows that yes, technology can predict better. And if you see the predictability, it goes up to 0.7% predictability. And one cow becomes grandmother in 70 months. But if you predict a newborn calf or maybe antenatal, then you are in a position to know the genetic merit and you are going to save at least 4 years. And for a unique time, rate of genetic gain will be 4 times even if your prediction is normal. We can't wait to a grandmother's daughter to get evaluated that how much will be there. It's so simple to understand that how genomics can actually accelerate this, and it has accelerated. But if you see, this is the interesting graph. Even I tried to convince this to our topmost authorities that this is the USDA graph. If you see here, very interesting point, that genomic selection started here, 2009-10, and cow genome sequencing was completed here. So a lot of SMP came today. You have 777,000 kg. You see the interesting part, number of animals is going down without compromising the genome. Very simple to understand. Like if the topic is really agriculture genomics, computational genomics is required in agriculture. So you can see here, if you see in terms of fodder, you see in terms of methane production, you see in terms of biological efficiency or feed conversion efficiency. Though I have never studied animal science or veterinary science or agriculture science, but it is very simple science to understand. You decrease my number of animals, but without compromising my desirable produce of the product, and this is so simple. But if you ask that can we translate this technology into our setup, perhaps I will surrender if I am honest for you. Now, if you see the 18 different countries, how they are managing, then performance recording varies from country to country depending upon your interest. Like you are interested, like if you see the Swedish country, they are much more interested in fatty acid type because of the cheese. So the texture of cheese, aroma of cheese, because that has a market. But if in India, if you see, then maybe we are interested in your somatic cell counting because mastitis is a very common disease. And then the total yield and the milk combination including the fat and protein, those things we require. So this also requires a kind of planning in what rates is relevant in your market so that investment can be done and that return can be expected using computational genomics. Now, if you see the number of samples for this kind of advantage using computational genomics in domestic animal, your reliability gets increased the moment the sample size is high. But if you see all your coordinated breeding program, you will never find this kind of sample size with phenotype or DNA bank, and this is the greatest thing we are struggling to convince our system. Now, I will show you the final result in only 2 slides. One is, this is the result of SFA chip and association study of GWS. These are the different fatty chromosomes of the cattle. If you see the bacterial disease like mastitis, we call it cannella root, where you get blood in the milk, and that's a very common disease. And if you see here, these are the 2 different chromosomes where you get SFP having symptoms with the association. So it becomes very easy to understand what kind of genomics is being backed in different breeds of animal or population. But at the same time, if you see the FMD, foot mouth disease, which is very common and it requires repeated vaccination, you may not find a clear-cut association. So for every trait, for your secondary or tertiary stage selection using agricultural genomics, it requires this kind of data condition. Then only you can move forward. So this is all about domestic animal. Now, if you see how far it is being used in case of say, fish, you can see the salmon fish, and it has its SNP chip is also available, and it is being used for 6 or 7 important traits like the feed conversion efficiency, body growth, and the disease and the feeding ability. And if you see in our condition, like if you see the total protein requirement and the expectation from the aquaculture, and if you see the program, like how far we are gearing up, maybe in terms of fish quality or fish production, related to the genomics, we are at the stage, like we are just sequencing 2 of the important fish supported by TVD. We are also one of the partnering center, but we are far from behind. Now, if you see the world status, one slide is just wanted to show you. Like people, they have gone for market rate association also for the important rates, and work is being. Now, you can, for knowledge discovery part, because we are much more towards the service organization as far as the ICR is concerned, but knowledge discovery, we should not underestimate. From here only we get the knowledge that where selection has to be applied. So, the genomics kind of project is going to crack the important traits in which we are interested. These are just like an example. So, in that sense, we wanted to see how far domestic animal the world has gone. Really, it is giving some most indicative genomics, excluding India, and we also saw some of the examples of fish and how it is giving a boost and how what kind of technology is available. Now, let us see very quickly crop. Then you can see almost all important crop. They have been sequenced or they have been used for SNP data generation, and the chip is available and it is being used in selection. So the latest chip is available is of wheat, which is very high number of SNP, 822K. We have been very fortunate to use this chip over 500 important wheat varieties. We have generated the data. I will be sharing some of the results, which now we are moving forward from our new center. Now, if you see the rice, your 3000 rice variety genomic sequence is available in public domain. If some of the researchers or the faculty from this sector is interested, at least 400 Indian rice variety genomic sequence is available freely in public domain on World Hunger Day by the Heath International Rice Research Institute, and that data size is around 18.5 petabyte, and that 400 belongs to India in terms of our interest, and those things can be further analyzed. So the data is not at all an issue. The only thing is right kind of team and the right kind of interest getting it geared. Very quickly, if we see in case of wheat, you will be really surprised that Pakistan has been doing better, especially Professor Qureshi group in Qaidar Azam University in Islamabad, and they have used the genomic selection using wheat SNP chip in 2011 to 2013 crop cycle, and they have the result. They are using it in terms of field using the SNP in progression. This is the reality that they were having a joint partnership with CIVIC, the International Heath Institute, and also with BGI. I am not criticizing, but I am just emphasizing. Sometimes we may not be able to do ourselves, but there is no harm in joining hands and moving forward. I am not appreciating Pakistan as a Pakistan, but as a scientist. Like we were not knowing how to handle the server. We went to Professor Raghavan, and he was kind enough. Then actually that gave a birth to the team, and we started moving forward. We faced so many questions that how CSIR can have an agriculture mandate or agriculture institute can have an mandate, but there was understanding and true spirit, and finally we learned. So that is the message I am trying to give. And there are several examples of different wheat varieties and its SNP and trade association. Let us imagine that okay, the world has done for a specific disease in wheat, specific trait in wheat, and their markers is available. Their rate of the genetic aid will be much more faster than the traditional breeding. Some of the important qualities like celiac disease in wheat or your chapati making ability not to wheat, and I will say that the 6th district of Ashoknagar in MP, where you find better quality wheat having a very unique cluster, and its price is much more higher and it is getting exported. If you see the oldest literature of Mumbai film industry, you will find that in the illustrated weekly, the internet, you will find some article saying that there were some brokers, they were taking these wheat and supplying to the Mumbai film industry at the time of London and Hema marketing, and still it was having health benefits. Those things we don't know. Still we pay higher price, and today we do not have a DNA certification of this quality wheat. And certainly very soon we are going to have, because we have used SMK's signature for this variety, but our paper has not come. So the message I am trying to give, it's not only the rate of genetic gain, it is sometimes DNA certification of the quality, bringing more income to its and less quality protection. Like Basmati, today we have 4 different DNA testing system. So the message here is wheat and SMP chip is going to give that kind of result which is actually required. Now this is the nutshell of the global status. We saw how it is being used in domestic animal, how it is being used in fish, how it was used in crop, especially for your all kind of crop. You saw the example and rice and wheat in detail, because that is our daily use agriculture thing which we feel. Now, when we started in 2012 after meeting Professor Heath that how to move forward, and we were not knowing that how to use global IP to develop a server, how to go for code logging. Probably you should laugh, you may laugh, but we feel honored that we landed in so much safer hand that he never criticized us that how we were taking free. Rather, he supported us that yes, these things are not very difficult. Go ahead and I am with you without any terms and conditions from the market. And today we have at least few servers working, and every server is now published, giving us the paper. And in last 2012 to this period, our group has published at least more than 20 papers in the public domain, and certainly not any one of them from India. We submitted at least reasonable journal, and we made a very humble thing. Now, you see some of the unpublished, which we are looking forward to get published very soon. They are in the pipeline. We have wheat variety identification system, and that is going to be perhaps world's first DNA-based prediction system for wheat crop, covering more than 500 varieties. And we are now going to have rice. Our server is working, our manuscript is under publication, and it is having 900 rice varieties, including the rare variety of rice, which was brought from Burmese in World War II in Port Blair and Andaman Islands. We have been lucky to have those kind of rare rice variety protected in sovereignty super IPM. So these are the list, and you will be able to see some of them, and I am sure when wheat and rice comes, it will be a national headline in this country, that we are very sure. But product is ready, we have not launched it, that we belong to commando force working. Our team wishes that, our team is wishing that first papers should come, then only we should disclose this. So these are the small applications we are able to get it, because we do not have enough data in terms of phenotyping. But now we have SMP chip data of wheat, we have SMP chip data of mithun, we have SMP chip data of your domestic animal, especially for cattle. DPT has supported us. So now we have made a very humble beginning, but the accelerated rate of genetic gain, it is not being done. Let me confess this. Now we started a humble beginning by our team in different area, small work, and here comes Professor Raghav sir, the summer of 2012, when I shifted from Karnal to Delhi, and we went to Professor Raghav sir, and we asked that, sir, we have more than 50,000 genetic data of DNA fingerprint of the Indian gold breed, and we want to put machine learning. We also wish to put it in a server mode, and we also wish that number of brokers will be reduced using the machine learning, developing the right model, and it has to be implemented, and it has to be user friendly. And the data should be fed maybe in base pair, or maybe your CSV file, or maybe your notepad. And in one or two meeting, I am sure, as Sir convinced us, and looking at his face, we were knowing that we may not be that big, but his face confidence made us to move forward, and finally we could be able to publish. And to my utter surprise, like one day night, I was unable to sleep in inferiority complex, that how this paper is getting published. And within 14 days it became highly accessed, and today it is world's #1 in the domain area in ranking since last 3 years. Every month we check its ranking, and the credit goes to the mentor. He is sitting here, and Professor Ramasan. And this is actually, we call it like program 1. That gave us the confidence. We started getting inflow of the data, and we started getting good collaborators, because little success is required to have super friends and good enemies. So we have been so lucky that we didn't get enemy, we got good friends. And a majority of them they supported, and today we have wet lab data inflow of 20 different labs from the country. And I feel like the 15 years back, the micropipette we just put it, probably what compensated us by making 20 different satellite labs and still get the data, and we trained too big. In fact, we wanted too much than what we could actually deserve. So immediately our next question was that if you use 25 locus to fingerprint to identify one brain out of 24 brain with 97% of accuracy, can we reduce the locus by using your ANN technique? Then, except me, everybody was a statistician or computer scientist. Then Professor Rava said, yes, it is possible, so you can move ahead. So using same kind of data set, we published another paper with animal genetics. And here the title itself says that locus minimization of breed prediction using artificial neural network approach. And this paper also gave us the confidence that the cost of genotyping can be reduced to 1/3, and 8 locus was good enough. That will be tried for that will also be reduced up to 5. But we are still waiting for more data of that particular species. But these two papers actually, because of Professor Ramasan, I tried to read that gave birth to team. I know for like us, he might have actually mentored few hundred, if not thousands of students, but you feel sometimes a great simple person can help even without having any official on-paper relationship. We just requested, he placed, and this kind of thing. So sometimes people put their hand, and then the iron becomes diamond. So that is their capability, and that is our fortune. Now, we started looking for computational genomics problem, because doing is one thing and publishing is different. Then we identified one problem, that if chromosome-wise markers are available in a sequence genome tracking region of the genes, our breeders can actually progress them. So we took the tomato genome, we developed the server, what we learned from Professor Ramasan, and this tomato genome paper, this was having some 4+ function implied. This also became in very same month highly accessed paper, and we were able to publish this, and our server is still working, and this can be used for therapy and identification. So I guess 5 minutes. So now we tried with several other genomes, and we started publishing the marker discovery, and we found an interesting problem, that can we resolve the genome assembly using marker development of PCR-assisted chromosome? We got the buffalo genome data from public domain. We applied this technique, and finally we published it in Games Genomics. So one thing we learned from Professor Ramasan, that if you have the scientific spirit, if you have a computational hand, and if you identify a right kind of problem, if you solve it maybe 10% or 20%, and if you are able to build a story, the age is yours. So we could publish series of 4 or 5 papers with 4+ function impact because of the Professor Ramasan, and that gave us the confidence. And by the time we started writing some random applications and the other collaborative sessions, and today we are flooded with NGS data. But the initial one was just because of the intellectual needs we required from Ramasan. So now we thought that can we discover polymorphism without putting the PCR by the sequencing genomic data and using ePCR analytic difference. So our team wrote the program, and finally we implemented it using ePCR, and this also we published. And then we learned that how to reduce the cost of wet lab, not replacement, but certainly cost of the wet lab. And when we published this paper in December 2000, at the same time Nitin Gadkari made a statement that sugar beet can be used microbiome. We don't know that why we did it, that it was just a coincidence. But we were surprised when we saw our work in Times of India and Hindustan Times. Nobody congratulated us, nobody appreciated us, let me confess this. But at the same time, our team felt very happy that yes, sometime our authorities also need some good work to display, to appease or to respect, whatever you call it. But sometime you become so popular that this work has been done. Since so it was used in sugar beet, after discovering plus it was a very hot paper for them. Now the plant variety authority is having a problem. 12,000 varieties is pending. We started working on it, and finally we are coming to the important paper that how to identify crop varieties. I will be sharing some of the results. This is the rice variety. India is having a lot of rice variety pending, and this is going to be world's first rice variety server where we can identify. This is for chickpea, and you see in China how they have improved the same technology. Like wherever the important genes are there, they are looking for SNP clusters, and using 9,000 SNPs, they could collect their 229 wheat varieties. And this is our internal result. You can see red color is your deployed wheat, and the green color is your green revolution, and the blue color is your pre-Mexican or our existing land-based or modernist wheat variety. So the message we are trying to give you is 35,000 SNPs in its data is able to differentiate, and finally this is the unpublished result. So 50 SMP is good enough to differentiate all important Indian wheat varieties in terms of its origin. Next. Now these are the small details of our projects, like this whole genome sequencing, first project of we are doing on, and it is going to be over this year. And we are also working on cattle, we are also working in crop. And Ministry of Agriculture has given us 68 crore project where 20 centers are there, and we are working for computational genomics. So the God has given us enough money, team, and opportunity, and we are moving forward. And the conclusion is genomics is the backbone of the solution, but we are at zero. The country needs to move forward. The greatest difference which I could see is, if you see the BGI where we are dreaming as a biopopulation to fight with, if you see the Executive Director General, its age is 36 years and 35 papers in Nature and Science. If you make a young person as a Director General of my sector, probably all of us will surrender. So this kind of mental impediment, I feel it's the greatest ever challenge I can think of. But Professor Raghuvar, a person like him, can transform the system. If he can transform in 72 hours to us, then till date we are inspired to move forward. So I think in national capital, sir, your presence will be really, I'm sure it is going to be an epicenter if we join and move forward. So I'm extremely thankful to my team. They made me learn as a biologist the entire bits of computational genomics. I may not be able to answer every computational question in terms of the script, but in terms of the spirit that why this script is written, I'm sure I will be able to respond at least some of them. Ask questions, and we look forward for heavy interaction with you. We have PG students, we have PhD students, and our PhD students are from IRI, and some of them are registered in other universities. So I understand because of the presence of Professor Rao, sir, we may have a joint academic family to move forward, and to getting Guru back in the same reason, I think it's a God grace situation. So with these words, thank you. With these words, thank you once again to each and everyone for this great opportunity. And India is going to get changed if Professor Raghuvar continues to stimulate scientists like us and students. Then definitely India will be no longer the India we used to see earlier. Thank you.
We have time for one question.
Sir, in a graph that we showed that the number of animals are decreasing, but the productivity is still high. So, is it also measured that the quality is still the same or is the quality decreased?
Yes, what you are asking, that's the most relevant question. Because the moment you increase the yield, you may have compromise in terms of quality. But here in this graph, particularly for USDA or even otherwise also, when you are using the SMP chip data, you have the precision phenotype of all important traits like production rate, then quality is never compromised. It's never compromised practically. But there is a possibility, like all high yielders, they are disease susceptible. So you give enough weightage. I will give you a precise small example to convince you. when you give 33% weightage to yield, like milk yield, 33% weightage to its longevity of the animal, and 33% its ability to fight the disease. This is called your Scandinavian model or mixed model or the best model. Then you are not compromising. But if you see the Israel model, 100% productivity, then some of your animal starts falling sick. So it is the weightage you fix, like how much you wish to take.
Okay, I guess other questions have to be offline. Let's consider once again. Small.
Thank you.
I'm really honored to introduce Mr. Raghava. We started the day with an exceptional mentor, and the mentor was able to train a mentee who turned out to be another exceptional mentor. Mr. Raghava has joined AAA Delhi just a few months back. Before that, he was in IIM Tech Chandigarh, and I've interacted with him only for a month or so. So I've noticed that he's very, very simple and he's very, very humble. You won't realize, I didn't realize while talking to him that of his abilities. He has exceptional vision. He believes, he joined IIM Tech as a computer developer, software programmer kind of stage, and he left it as the head of the Bioinformatics group there. And he has a wonderful vision. He believes in open access to knowledge, and that knowledge should be free, and everyone should be able to use it in the best possible way. He has led a lot of research activity based on that, and his team has developed lots and lots of web servers that are used consistently, and they're highly used all over the world. And it has earned him a lot of accolades. He has got the fellowship of Indian Academy of Sciences, National Academy of Sciences, and he's also got the Chancellor of this for a few years back. So let's welcome Professor Raghava.
Good afternoon, everybody. So everybody is waiting that it should be finished fast, so we can go home. Anyway, I will try to. Thank you very much for listening to my work. I have done a little bit with So it's interesting. In this age, I am feeling younger. Why? Because I had just joined 2 months back. So everything is new for me, and I am asking from everyone that what is the procedure here, how it works, how to organize the workshop, all sort of things. I have nearly no knowledge of this place in last 2 or 3 months, whatever I have learned. Number of persons ask me this question. You are leaving the institute after 30 years actually. So how you feel? For me, it's perfectly alright. Perfectly alright in the sense that whenever you change yourself, you always learn. Change is very important in the life. Most of the time, we protect ourselves, we don't want to go out of ourselves, we want to keep in the self. Till you talk to the persons, till you talk to the other people, till you understand the other people. How do you understand the problem? Dr. Grace just mentioned that I help them actually. No, it's not the fact. I learn a lot from them. Why? Because they come to me. I talk to them. We discuss them. We are collaborators. We discuss a lot of things. So they also give something to me. I also give. And I want to give the statement, if anyone is interested and want to work with us, you are most welcome. And the fortunate part here, bureaucracy is not there in the institute at least. So there is no problem, you can come anytime, you can discuss with me. You never feel that I am busy or whatever. I am not busy. Because I have the full force. You know full force? They are sitting in the back. Means lot of manpower I have. That's not a problem. I always kept time for myself. If I am not happy, if I am in the tension, if I am too busy, how can I think about science actually? You cannot expect good science from a sad person. खुद दुखी है, क्या साइंस करेगा? अपनी तो भला कर ले पहले, दुनिया का तो भला कर ले. So I have the simple theory, पहले अपना भला करो, बाद में दूसरे को भला करो. It's very simple. So I have sufficient time. I'm a happy person. And if you need any time, any help, you are most welcome. There is a reason for that. When I talk to anybody, I also learn more. Because you will tell your problem, I will try to learn it, or I will read it. So in this business, we both learn. And our field is informatics field. Informatics field, we are not doing any experiment. We are not doing any biological experiment or not doing any physical work. What is there? We are just taking the information, we are processing. So group here, in CD group here, in this issue, I talk a lot actually. I disturb them a lot. I force them to talk, to learn and all. It's a deliberate. Means we have a more interactions, we can talk the science and all. It's important in life how you think. That's most important, how you think. You think in the long term or you short term. Short term in the sense you take work from somebody and then if you don't need the person, then you are not bothering about the person actually. That's called short term success. You are bothering about it. Long term success, you do whatever the best you can do. And you will get in return after some time. This is fact. So whatever I have done, I am not claiming any science or I have decided 30 years back when I started developing the first software that I will give free to the public. Whatever I have learned, whatever the knowledge we will get, we will give you free to the public. Though there will be competition, it's fine, no problem. We have to fight tough and all for it. But if others will use our softwares, data sets, and other things, then they will naturally accept your work. If you will hide everything, so you will die with the knowledge. That's a major part of this country actually. We have a lot of knowledge over the time, there is no problem basically, but we have not documented and we have not shared it because due to the infinity complex. We are not confident on ourselves that if another person will learn, then I will lose my position or whatever. Anyway, so otherwise it will go the big lecture. I am not going to. I will just tell application of genomics in health. I am just covering few examples. So it's not practically possible to cover everything. So I will just give the few examples. Maybe it may help you in future. So these are the terminologies. I am not going to talk about omics, all omics means let's say genomics, proteomics, epigenomics, glycomics, lipidomics. There are different good terminology are there. So they are quite heavily used. So if you are the new person in the field, you just remember this terminology. When you talk about the, let's say, for example, about the personalized medicine or let's say person-to-person variations, how you will think? One is DNA sequence. It is possible that two persons have the different DNA sequencing. So there is a mutation associated, disease associated mutations. So the mutation in the genome will affect it. So that is one possibility, that DNA sequence of different persons is different. So we have to understand that part. So that may help you to understand the person gene. Another is gene expression. Gene expression in the sense, you have let's say 20 to 30,000 genes. These genes are expressing real life. There is a different expression of different genes. You can measure the expression. Microarray data is nothing, it's just related to the gene expression data. So you can simply take the cancer patient or a healthy individual and then compare and find out which genes are more highly expressed in case of the cancer than the normal differentially expressed. So this way you can find out the biomarker, or maybe you can see the drug effect. Let's see if you are giving a particular drug, which gene expression will be affected. If it is affected, whether it will give side effect or not, that depends. So that's another important part. Another is copy number variation. Copy number variation, in the sense, so you consider 20,000 genes. For each gene, not necessarily only one copy is available. There is a possibility that multiple copies are there. And sometimes this multiple copy also correlate, not 100% correlate with gene expression. And the advantage of copy number is, because gene expression is a dynamic variable, which is changing with the time. Maybe you take a sample from me now, and you take, let's say, even after food, it will be different gene expressions. Okay? But copy number variation will not change. So predicting gene biomarkers and predictive sort of thing, it is better to take the copy number variation, because that's a more static part. It's a part of the gene. And single nucleotide polymorphy, that's the part associated with the disease. These are some examples. And we call this field pharmacogenomics when we connected the genomics with the pharma, means effect of the different drugs. So some people are practically FDA approved. There is a number of biomarkers in there for drug biomarkers. What is drug biomarkers? They simply check whether the particular drug will work or not work, whether for this person this drug is effective or not, or whether it will create some side effect. So there is a number of biomarkers. This side there is medicines, this is a disease, this is a treatment decisions. Based on the certain parameters, they take the decisions. For example, there is a Herceptin antibody. We only give that antibody if HER2 expression is more. If HER2 expression is not more, EGFR expression is not more, well, then it's not effective. So there is already biomarker, you can save a lot of cost as well as side effects. So this is just an example. How we come into the cancer business. It was in 2012. A paper was published in the Nature where they study of 900 cell lines of large number of drugs, and then they check IC50 value of each drug, and then they sequence the 900 cell lines, not all sequence, all genes, around 1600 genes basically, and expression of all the cell lines. So this was a good starting for us, because we want to start this field. We have no expertise before this time in our group, so we started at this time. What we did basically simplistic, because this is important for the youngsters basically, who is starting the career in bioinformatics particularly. It's not difficult. You should be a little bit smart. I will tell you, in biotics, if you want to start your career, easiest thing is make the database. Make database. What is the advantage? There is no science behind it. You are just collecting, compiling the information. And if you are lucky, you can get the publications in publication research, 80 factor is there, okay, or database or crossword. So that's not a big deal at all. So that's the easiest way. In biotics, if you are starting the career, you just make it one database. And that's a sure shot, it will publish if you are able to collect and compile the good information. Now you will ask me the question, what is a science? I want to do a science, and you are telling me the routine type of job. Okay. Advantage, if you create a database, you read all the literature related to that. So you get depth of the field. If you really make the database of any field, you have become the master of that field. Now you have the data with you. Now you can think to design some prediction methods using the data you have created yourself. You have the advantage because you have created this database, you have the depth knowledge of it, and you have the complete database. That's the advantage. These foreigner people have that. They have the NCBI, they have the EBI, DNA database. There are a number of databases there are all over the world. So we are just Indian are just using them. We have not thought about creating the primary database. So we have to change the concept. If you give the fundamental information to the world, then you have a better. Okay, so we create this database, lot of information is there. It's nothing actually, just to collect and compile information available in that nature paper. Lot of material was there in the journal itself actually. We collect and compile, and we did some extra work like structure annotation of proteins and all. So that was our contribution. So this is just example how many fields and drugs. 148 drugs die on 952. So you may understand that how much data will be there. Okay. So next challenge for us was on Herceptin. Herceptin is a quite costly monoclonal antibody. Per dose is around 3 lakhs or something like at that time, and 3 or 4 drug dose is required. So person is spending around 15-16 lakhs, and there is no guarantee that it will come. There is only one cancer biomarker that's based on the EGFR to overexpression, HER2 overexpression, and that doesn't give the 100%. So what we did basically, we collect and compile the information from literature and put it here, so you can design based on the genomic information whether the particular drug should be given or not, or if it is given, what should be the supplement. So that information will be used. This was another example because at the same time there was another report in Nature paper that why we are not able to control the cancer. According to them, they take the 1800 tissues, okay, and then they cluster it, and they found there is 10 type of breast cancer. 10 type of breast cancer, and present drug is only work for the one type of cancer. So we fail because we are unable to design the drug for other type of cancer. Question is, can we design a drug which can work to different type of cancer? So here we have made an attempt to design a QSR model for pancreatic cancer cell lines. So this. Please remember one thing, we are just starting. We are not claiming that we have done any great thing for the pancreatic cancer or anything. Please remember this thing, I want to clarify. Because this work is on cancer cell line. Not necessarily it will work for the real patient. This is important, that whatever work we are doing, we should know the limitation of that. But it may help you. It may help to reduce it to many number of clinical trials. Instead of taking the all drugs, take only those drugs which work against the large number of pancreatic cancer cell types. So the chance is there, success will be more. This is another, that's why I was just joking in starting. Publishing number of papers is not a problem. I will give you a good example. You take that Nature paper in 2012, publish that paper Cancer DR. Okay? This is another example from same data actually. Nothing actually. What we did basically, we just take the data, we find out the correlation between the drug and gene expression. Means if the particular gene is expressing more, then this particular drug will not work, or it will work. So this server is very interesting server. It simply says prioritization of anti-cancer drugs. Let's say you have the 24 anti-cancer drugs, which is FDA approved. It If you go to doctor, what doctor will do? Will try hit and try this 2 or 3. If this is not working, try another 2 or 3. What we are saying, take the sample, do the genome sequencing, predict from our sample, and it will rank the different drugs for this type of gene expressions. This drug will be best, then next. So this is a simply prioritization of anti-cancer drugs. So we are not doing any clinical trial or anything. This is already well-known drug. We are just saying that based on genomic information, you can prioritize them. So I don't know how many people will use in real life, because why we don't know that information. Because too many people are using our servers. Some people give us the credit, some people doesn't, and particular company will not give the credit. We don't care as far as it is helping the public. So this is an example. This is another example. Same data, another paper. I'm just telling you the tricks actually. Okay. Because 900 cell line, we know their genome sequence. Now we compare sequence of cancer patient with the human 1000 genome, which is healthy patient persons, and then we identify the mutations which are cancer specific in these cancer cell line, which have not been seen any time in human 1000 genome. It means they are cancer associated mutations, right? Now take this region which are cancer associated regions, just check it whether they can activate our immune system or not. Whether they can activate our immune system or not. If they can activate the immune system, means B cell, then they may work as an immunotherapy. You can produce the antibodies against these peptides, and then you can use for the vaccine. Or same peptide you can use for the vaccine. So we are not doing any great thing, very simple sort of things. But if you see logically, they are practically useful. So this is a way, first time we try to predict the epitopes against the cancer cells. Then we also have the partially personalized concept of the genome. What is this partially personalized? According to us, take the tissue of the affected person, cancer person, do the genome sequencing, compare it with the 1000 genome sequence. Compare it with the 1000 genome sequence. You will get the regions which are cancer associated, which have not been seen in the normal population. Then check it whether if they can activate the B cell, F2 or T cell. If it can activate, then they may work as immunotherapy or vaccine. Am I right? So simple. Another label, fully personalized. What you have to do? Simply take the, let's say, healthy tissue of same person and cancer tissue of the same person. Do the genome sequencing, compare it, find out which regions are different, and then. So this is a perfect example of personalized immunotherapy or this is the new database has been created. That's again good thing actually. In our group we are always creating the new database. It's nothing, we just collected and compile the information about the proteomics data, MS data, proteomics data, the peptides found in the body fluids, different body fluids. So we have just published this. Now we can take this data just to find out the biomarker, peptide-based biomarkers. Because we can just check it. The person, let's say cancer person, have this type of peptides released in the body, which is not seen in the healthy person. So in the future, people can use this database for designing the different type of biomarkers. It may be blood based, or it may be urine, or whatever the body is. So this is just recently published. This is another, I think in the morning already Dinesh talked about the classification of the different stages of the cancer basically. So we have taken his data, his information, we have worked further it, and then we proved that method. This is just example of the algorithms I'm recording. That's another part of the, if you are working ADMET, then how it will metabolize, which type of isoform you have, then you can use which. This is an example of HIV. In HIV, how the HIV will enter depending on that what is the sequence of the particular gene. There is two things in the personalized medicine. If you are talking about the personalized medicine, I have the two concepts. One, for your self-use, because every individual is different, so you can think about the personalized. And another part is strain-based vaccines or drugs. What is mean by that? If you are infected by any particular disease like HIV, TB, or doesn't mean that all the people are infected from HIV. They are infected from the same strain. There may be variation in the strain. So there is a different strains of HIV itself, same MTB itself. So if you give a drug or vaccine, strain is the same. That is also got some type of personalized. So in this case, based on the sequence, we can find out whether it will use the CCR5 receptor or CR4 receptor. Based on that, you can use the particular drug. This is the example of MTB. What we did here is to take the MTB different strains and we give the concept that how can you use the genome sequence of MTB for designing the vaccine. It may be peptide-based vaccine, it may be antigen-based vaccine, or it may be the whole genome-based vaccine. We have given the concept there. This is just jumping. And sometimes you have to work for the real-life problem. So recently there is a problem of Ebola actually. So we have not done anything, but we just collect and compile the information, genomic information about Ebola, and based on the prediction tool, we just predict what will be the possible vaccine, what will be the possible SRN-based therapy, and different types of drugs. And we are not claiming that they will work in real life. You have to test experimentally. But it is an advantage for the experimental biologists that they are not supposed to do the experiment. Means biotics, they will get the data from real life. Same for the Zika virus. We have recently shown. This is a big team actually. So whatever the work I am just talking here, it's easy for me to talk and to say that we have people alive and all sort of things. But ask me by students actually. How it is easy with me. Okay. So a lot of tension they have. Thank you very much.
Any questions? Sir, I need to ask, is there any database by which we can predict antimicrobial activity by seeing the carbon score of the molecule?
Yeah, there is two things. One is antimicrobial peptides. Antimicrobial peptides. There is a server for predicting antimicrobial peptides. So if you are interested in that, there is some is based on small molecules as well. But they are for particular type of assay, a particular target. Peptide.
Sir, is there any database for the cross-reactive disease?
What?
Cross-reactive disease.
Give the example.
Like the patient which is having a TB,
Okay.
most commonly is having HIV also. But in case of leprosy, the HIV is very rare.
I have no idea. Anybody have any idea that there is any this type of database? I have no idea.
Any other questions?
In morning there was a talk about epigenomics. So I think that epigenomics is a more clear picture of the genome because it is very what are the activities and things like that. So in treatment of the diseases, why don't we compare the whole epigenome instead of just the DNA?
That's interesting. I will say these are different beautiful names actually. I will say simply these are beautiful names. Because when you talk about gene expression, that is as a dynamic part. Okay, so these are the big terminology. Otherwise, we are considering everything.
Other questions? Okay.
भागने का पड़ा होगा, सबको भागने का पड़ा होगा। ऐसे टाइम में कभी प्रश्न नहीं पूछते। मुझे दे लो। ये तो इंटरेस्टिंग है कि ऑर्गेनाइजर को।
Raghav sir, especially for you sir. I know you since last 7-8 years. It is the first time I am listening in face to face, and I am really a good fan of you because many of my students and my batchmates work with you, and I have almost every information what you did in last 5 to 6 years, means what publication you have, what databases you generated, everything right from scratch to the final publication. So I am really a good fan of you, and I appreciate your work, and you are a leader of and inspiration for many of us, including me also. Thank you very much.
Okay, so this is a concluding session. We will not take much time. I hope everybody have fill the feedback form. Anyway, decide filling the feedback form क्योंकि इंडिया को लिखता नहीं है, डरता है कोई, पता नहीं कौन सा लीगल एक्शन हो जाएगा. Right? If anybody have any suggestions for this work, we are planning to organize more workshops in future. This is first workshop after I joining. For me, it is the first workshop. So I want to know if there is anybody, any feedback, any suggestions on the workshop you want to improve, or you feel there is some shortcomings in the workshop. It's not this symposium. I will. वो तो लिख दिया होगा अपने फीडबैक में, तो उसके बाद भी आप कुछ बोलना चाहते हैं ना, जो लिखना नहीं चाहते, बोलिए। तो ये हेल्प करेगा हमको फ्यूचर में मतलब ऑर्गेनाइज करने का, या आप फ्यूचर में किस टाइप की वर्कशॉप, कॉन्फ्रेंस, सिम्पोजियम यू वांट टू सी एट दिस। कभी नहीं आओगे उसके बाद बहुत परेशान करते हैं। Means you are talking about the tools. R is a part of it. Anybody have any suggestion for future?
Sir, I have one. In future, if one workshop, we can have in-hand
something like 1 week workshop, or something like in the morning there is a lecture, in the evening there is some hands-on actually. Then you will learn the practical.
And it should include the statistical analysis as well as some software.
Right. Then you will learn. One thing I want to say, if you are a biologist, and if you are scared from the mathematics or software, you should not worry about it. You are just using it. So using any software is not a big problem. You can use it.
Some workshop for computer background students like basic knowledge of biology.
Whether the computer scientist will be interested or not, that's it. How many people are computer background here? Anybody interested to learn biology? Maybe we will. Important. But both communities should come together. Anyway, if there is no more feedback, then we'll have the group photo. How you will make the group photo? सबको बुलाएं, आ जाएंगे आप? आ जाइए। बचपन से देख लो, कोई स्टेज मिलती है ना, आजकल जो भाषण दे रहे होते हैं जैसा उधर पड़ती है पूरा। ये बंद करवा दो। हाँ, कुछ नीचे आ जाते हैं, कुछ ऊपर आ जाते हैं। कंपनी में तुम्हारा विनोद आ जाओ, पीछे बैठ के सुन रहे थे।